Ciao. we can start. If everyone's situated, comfortable, good. Okay. So typically, our executive director, Jeanette Schnars, gives the welcome. This year, I'm doing it. She's not able to be present for the opening session. So I guess I would like to say, first of all, welcome to the Regional Science Consortium's 19th Annual Research Symposium. So I'm Jen Salem. I feel like I know a lot of you in the audience, but for those that I don't know, welcome. I'm the plant manager and greenhouse manager. We do have the consortium headquartered here in the building. So if this is your first time here, again, thank you for coming out. We operate from the research wing here, that's where all our labs are located. And our little lean-to greenhouse, there's nothing in it right now, so I apologize for that. But we are made up of 36 member organizations, and they include colleges and universities, state and federal agencies, and school districts and other nonprofits. We support a mission of scientific research in our community, and particularly in this region. During this time when scientific research is more important than ever, we are excited to host this symposium. So over the next 3 days, we have a great number of presentations, including 39 oral presentations and 53 presentations given by researchers, faculty, and undergraduate and graduate students. In addition to the in-person lectures today and the rest of the week, we are live streaming everything on the Regional Science Consortium's YouTube channel, and that is on RegSciTV.com. So, and thank you, Eric, for being here and being our tech support today. We are inviting you also to join us tonight for our poster session. It is here in the building. It is from 6 to 8 o'clock, and students will be here to present their posters. There's also complimentary beverages and food. And then we also have an after hours on Thursday from 6 to 8, and you're also welcome to join. Then we'll have a live band. It's the Sam and Frank band, if anyone is familiar. They're great. So it's worth coming back just to kind of talk to other researchers and kind of catch up on what's going on in the scientific community here. And if you have any questions, please feel free to ask either myself or anyone at the registration table. So we have Haley, who's here, as our education and outreach coordinator. And then we also have Sean and Sarah out front. So please make yourselves at home. We look forward to kicking the presentations off. And I am moderating this first session, so you're stuck with me for a little while. And I think if we could have our first presenter come up, I just want her to check her presentation, and we'll get started in 2 minutes. So, do we have Kira and, let's see here, Delia? Adela, sorry. Don't have my reading glasses with me right now, so my bad. All right. So you have your stuff. Okay, so your presentation is loaded here. Yes. And then you have, this is your laser pointer, and then advance and back, just so you know that. Okay. Alright, so your presentation looks good. We just need to get it over here. Alright, I'm right, our first speakers will be Kara Armstrong and Della Mamani, thank you. A census of bat population of Gannon University in Erie, PA. Okay, I'll let you two take over.
All right, good afternoon, everyone. My name is Kira Armstrong. I am currently a junior at Gannon University,
I'm Della Mamani, and I'm a senior at Gannon University.
and we are presenting a census of the bat community on the campus at Gannon University, Erie, PA. Now let's talk bats. Bats are the only mammals that are capable of true flight. They are considered mammals because they give live birth and produce milk for their offspring through mammary glands. They make up about 1/4 of the mammal species with 1,100 different species of bats. They can live almost everywhere in the world except for the extreme desert and polar regions. Their bodies can't handle the extreme hot or cold. They feed at night and sleep during the day, so they are considered to be nocturnal. Even though they pollinate flowers, disperse seeds, and eat millions of insects, they have been scientifically neglected until recently. Bat numbers are decreasing because of a new disease affecting hibernating bats called white nose syndrome. Now, what is white nose syndrome? We also call this WNS, so throughout the presentation I'll be referring it to that. This is caused by a psychrophilic cold-loving fungus. It lives in caves since it provides damp and cool conditions that allow for optimal growth for the fungus. Over half of the U.S. bat species also hibernate in caves, so the WNS will manifest on the bats. The fungus usually manifests on the face, ears, and tails and for extended periods of time, so basically it could stay in the caves and manifest in them for a very long time. If you can point. All right, so as you can see, the WNS has manifested on the bat's face, ears, and a little bit of the tip of the wings. And on the bottom picture, you can see that it manifested on the bat's wings. In this very large picture on the right, you can see that it manifested all over the bat. WNS causes bats to wake up during hibernation, using up their precious fat reserves, and it can also suppress their immune system. So imagine you have very painful while you're sleeping. You want to scratch yourselves awake, right? It's very scratchy. So they'll wake up and then they'll use their fat reserves. So they'll have to leave the protection of the cave to go replenish those fat reserves. And then once they leave the cave, it's kind of during winter, so the weather conditions will actually cause them to freeze and have hypothermia and die. Mortality has approached 100% in some caves. Since its discovery in 2006, WNS has already killed over 7.5 million bats. Now that's a lot of bats. Scientists are now saying this is the most precipitous wildlife decline in the past century of North America. That is a very big shocker. That's why we added this quote, because this is a huge decline in the bat population, and bats are very important for our ecosystems. Before I play this, this is a map of the current WNS status. It goes from 2006 all the way to 2022. This red X over here is the start of it in 2006. You can see that it gradually started to increase on the eastern part of the United States. It starts to make its way up into Canada and starts going to the southern part of the United States. Around 2016, you can see a jump all the way over to the eastern part of the United States. All right, let me stop there. If you can see from that red X, from 2006 all the way to 2022, that is a very large spread of WNS in our United States. Concerns. There are two main concerns that we have to worry about for the bat population if their numbers continue to decrease. Extinction. 9 species have already been affected by WNS, and 4 are federally endangered now. The strategy at this point is management and conservation of bats and their habitats in the path of WNS. Trifecta of environmental, agricultural, and financial problems. Now, envision with me. So, if the bat population continues to go down, there's going to be a rise in the insect population because bats majorly eat insects. So if the insect population continues to go up, farmers have to worry about their crops and produce. So they're going to have to invest more into pesticides so the insects don't eat the plants and produce. But that means they're going to have to invest more into it, so financially that's going to be a problem for us too. Since we want to buy the produce and the plants from the grocery store or the market, more for the produce, which should only be a few dollars, but now it's going to increase because of the pesticide problems. Treatment for WNS. Researchers at Georgia State University have developed an antifungal treatment using the bacterium Rhodococcus rhodocraus. Rhodococcus rhodocraus has been shown to slow mold growth on the ripening fruit. In 2015, 75 bats were treated by hand with the bacterium and were released back into the wild before winter, and they survived. This is an amazing progress that we have been shown, since usually a bunch of the bats will hibernate, wake themselves up from the scratching of WNS, and they'll go out into the winter weather and they will die. But showing that they have survived is immaculate progress. Researchers are continuing to work the Black Diamond Tunnel to see if they can find more treatments for WNS. Further investigations. Bat Conservation International is collaborating with researchers from Western Michigan and Ball State Universities to test a newly developed antifungal agent, chitosan. Antifungal volatile organic compounds known as VOCs, essential oils, and other compounds are also being investigated to see if we can resolve WNS for bats. Development and testing is underway on a device to modulate the dispersal of these compounds to see if we don't just have to hand pick bats and give them the treatment. Instead, we can just spread them out throughout the entire cave and hopefully throughout the whole entire bat population.
Okay, so why is our location in downtown Erie, PA so special for these bats? Well, if you look at the map on the screen right there, you can see that we are directly across from Long Point, Ontario, which is in this little red circle here. And this is where these bat species that we're looking at will migrate to and from. So that means that we're not only located from the shortest distance across Lake Erie from Long Point, but that we're also the first stop for bats migrating south to replenish their nutrients, and we are the last stop when they travel back north to build up their fat reserves. So pictured here is a map of downtown Erie, and highlighted in yellow is the route that we take every day to look for these bats. And this features buildings and landmarks between West 6th Street and West 10th Street. And this route is walked every day from April 1st to November 1st by our student researchers and our faculty, Dr. Ropsky. So when we take these walks, we make sure to scan all the buildings that we pass to make sure if we detect any roosting bats. And when we do happen to see a bat, we want to make sure that we record what building they're on, what side of the building, so either the north, the south, east, or the west side, and how many there are, if there's more than one in the group, and what species we think they are. And for any bats that are out of our reach, that's the extent of the data collecting that we do. But if we do come into contact with a bat that is close enough to reach with a ladder, we're able to mark them with a dot scheme of washable non-toxic paint. And that way, when we walk our route the next day, we can see if that bat used the same roost the night before, or if they moved on to a better roosting location. And so while we're allowed to mark them with this dot of paint, that's as far as we can go in terms of actual contact with the bat, since the Game Commission has very strict guidelines on handling these bats. This is for multiple reasons. One of the big reasons being disturbance. We don't want to disturb the bats while they're trying to conserve their energy throughout the day. Second, we want to protect ourselves from any rabies that we might get from them. And then third, as Kira mentioned, the fungal spores that cause white nose syndrome are viable for extended periods of time. So if someone were to handle a bat with white nose syndrome, those spores would then transfer onto the handler, and if the handler were to touch other bats, they would risk infecting them as well. So here's a picture of a few of the bats that we were able to mark with this non-toxic paint. And our hypothesis for this research is that the number of bats in the Erie area would decrease from the previous years due to the presence of white nose syndrome. So starting off, our data is from 2012. This is just marked versus unmarked bats, and we were able to record over 1,260 bats, with a majority of them being unmarked, but still a good amount being marked. But then once we get to 2013, this is when we start to see the effects of white nose syndrome, as only 135 bats were recorded, but then the majority was still unmarked. And then this was pretty consistent over the next few years until we got to 2020 when only 7 bats were recorded. But this time, the majority of them were marked. So 5 out of 7 were marked. And then moving on to 2021, still very few bats were found. And then by 2022 and 2023, none of the bats we found were able to be marked. So now going back to 2012, we look at the distribution of bat species we're able to find. The majority of them were the little brown bat, along with the big brown bat, the tricolored bat, and the silver-haired bat. And then moving on to 2013, still same trend as the little brown bat was the majority, but now we see the introduction of the red bat, and there's no silver-haired bat for this year. And then there's still the big brown and the tricolored bat. And then moving on to 2020, the big brown bat was actually the majority of the bats that we found this year. 2021, we see a reappearance of the red bat, and the little brown took the majority for this year. 2022, the little brown bat was still the majority. In 2023, we had a little bit more of an even distribution, with 2 little brown bats, 2 big brown bats, a red bat, and a silver-haired bat. So now we go back to 2012 again to look at the number of bats per day. And what's important to note about this data is that there is a peak starting around the end of July, which is actually when baby bats learn to leave the roost and learn to fly, up until the beginning of September, which is when bats start to build up their fat reserves again for hibernation. But then looking at 2013, which I mentioned before, is when we saw the effects of white nose syndrome. You see a few peaks around the middle and the end of August, but not anything as big as 2012. And then as we get to 2020, we were finding so few bats that none of the data that we had for any of the bats we found was very significant in terms of any peaks. And then as we get to 2021, we had the same situation where we were only finding our very few bats every once in a while over the months. And then 2022, we weren't even finding any bats during August, which is when we said that they were starting to leave the nest to learn to fly. And then 2023, we were only able to find half our bats throughout April and May, and then throughout September and October. And then here we have a compiled data of all of our bats that we found, but distributed by building. And as you can see here, the most popular building that we found bats roosted on was the north side of our old main building on campus, followed by the east side of our Palumbo Academic Center, and then the east side of the Communication and Arts building. So we're going to look at why these places are so popular for bats to roost on, starting off with Old Main. If you look at the structure of the front of Old Main there, you can see that it's made up of this rough textured brick, and that's good for bats. It makes it easier for them to roost on, so they're not sliding off. And then you can see the ceiling in the doorway is a little higher up, so it lets them be in a more secluded area, and it protects them from the sunlight and the wind. And this is great for thermoregulation for bats. And then pictured here attached to the old main building is this iron gate. And bats have become accustomed to this because a lot of caves throughout the U.S. have now installed these gates to protect from the spread of white nose syndrome. And then there's a similar situation with the east side of the Palumbo Academic Center. You see this rough textured brick, and these ceilings are higher. It's more secluded from the sunlight here and the wind. And then you can also see where the bats would be able to roost up here. And then looking at the east side of the Communication and Arts building, still the same rough textured brick with little areas that are more secluded from the wind and the sun that bats are able to roost on. And then another area to mention that wasn't as consistently popular with bats, but is still a great site for them to roost, is this stairwell on the east side of our old main building, which we do refer to as the cave because the features are so similar to a cave, because this area is even more secluded than the other areas I mentioned. So they're able to roost up here, and they're more protected from the sun and the wind. And sometimes when it does rain, the rain floods this area right here, and it'll bring any bugs that it has along with it. And that means that the bats don't even have to fly out to get their food. So in conclusion, we found that over the years, the amount of bats we were able to find has continually decreased due to white nose syndrome. But there is a chance that bats might be making a comeback, especially in Pennsylvania, in Bucks County and Knockmixon State Park. And additionally, researchers at Michigan State University were able to discover bats that were resistant to white nose syndrome, and they were observing that the bats that were more capable of fighting off white nose syndrome were actually heavier in size compared to like the average size of their species. And along with this, they were able to find bats at alternative roosting temperatures compared to what was previously observed. And they're thinking that at these alternative temperatures that the fungal spores are not able to survive. And so for our future plans with this research, we do intend to continue it into the 2024-2025 academic year. And we will continue to be a bat-friendly campus, and we'll do this through making the community aware of where these bats plan to roost and what to do if they do happen to find a bat. And then we also plan to implement alternative roost sites, which are pictured here on the right, so that bats have more areas to stay in safer locations. So we'd like to thank all of these awesome people. So our professor, Dr. Rovsky, our biology department at Gannon University for just allowing us to do all this, and then this long list of great people that were able to take time out of their day to do these walks and look for bats. And here are the sources that we're able to use. Any questions? Mm-hmm.
I was just curious, did you see any bats that actually had the white nose syndrome? And if not, what variables were you looking at that could have led to their fatality other than white nose?
We weren't able to like definitively check if they had white nose syndrome because we're not allowed to handle the bats. And most of the times, especially this year, we weren't able to mark any because they were so high up, so it wasn't easy to tell if they had them. Mm-hmm.
The 4 or 5 species that you guys have encountered, what's their overwintering habitat and location? They're not overwintering on campus, right? Where are they going and overwintering?
So at Presque, we have the Eagle Point, I believe. So they'll migrate from Canada to Eagle Point, and then they'll continue to migrate down south. So none of them really actually stay and hibernate here, as I believe. They're usually just kind of relocating in a way.
Mm-hmm.
I have a couple of questions, and I'll make sure I keep it to just 2. Do you have any interest in doing acoustic surveys as well as the visual ones? I think you probably are missing some of the migratory bat species that would not be likely to be hanging on the side of the building. I know you encountered like eastern red silverheads occasionally, but they're more likely to be roosting in foliage or cabins and trees or something like that. So I think acoustic survey could be really good for bats.
We haven't really looked into that, but that would definitely be an interesting aspect to do more research on. So.
And then my second question would be about the migratory connection over Lake Erie. Do you have any literature on that? Because I'm really interested in the migratory pathways of that, so I'd be interested in learning more about that.
So from my knowledge, I can't actually say a full thing about it, but the migration path is it's just from Canada to Presque Isle and continues down. I would also like to know where exactly they like to stay at and why they move down through that path. It's mainly to replenish their fat reserves and stuff. But that is a good point. We should probably look into that more. Yeah. Thank you.
Wonderful, thank you very much.
Thank you.
Okay, I'd like to introduce our next speaker. His name is Jonathan Townsend. This is another presentation about bats. This is habitat sustainability mapping using logistic regression analysis of long-term bioacoustic bat survey data set in the Casadega Creek watershed.
Very
Nice.
well done. I have to put as many words into it as I can.
I was
It's
gonna
not good
say, I
science.
have to squint down to better vision right now. So thank you, Jonathan.
Well, thank you very much for that introduction. So this is my second or third presentation here at the annual research symposium, so I'm really happy to be here. And I'm excited to present on one of the papers that has come out of my PhD research. I am a fifth year, going on infinity year, doctoral student, candidate at the University at Buffalo, where I study bat biogeography. And so my project that I'm presenting on today is about habitat suitability modeling in the Casadega Creek watershed. Let's see if I get this right here. I'll just use the arrow buttons. All right. So really happy to have another presentation about bats, so I can kind of maybe gloss over some of the introductory material for everybody. But you should know that bats are in the order Chiroptera, very diverse order of mammals, the only true flying mammal with bat species present on every continent except Antarctica. And you might be wondering why you want to study bats, well, other than being incredibly cute little creatures, as the prior presenters mentioned, they're keystone species, they're indicators of biological health, they're apex predators, and they provide a wealth of ecosystem and economic services. And I don't know if this remote's working for me, if I'm just too stupid to work it. That's okay, though. Anywho. So just really quickly, we can talk about some of the services they provide, such as pollination. And I know this isn't a local species of bat. This is the orange nectar bat from Latin and South America. And I love this picture because it really nicely illustrates the coevolution that you see between bat species and the plants that they depend on. They actually have a special term for it. It's called a chiroptophilous pollination syndrome, where the plant species and bat species evolve so tightly together that they can't exist without each other. And if you look at the little pollen-bearing structure at the top of the screen, it is curved. And it's curved in just such a way to bounce on a bat's forehead when the bat comes to feed on it, so that every other plant that that bat goes to then becomes pollinated. And this can help not only with pollination, but with genetic diversity, because bats are flying around and they can travel long distances in the evening. Seed dispersal is another important ecosystem service where, without getting into a lot of the details, just one example with Seba's short-tailed fruit bat, where a colony of just 400 individuals was estimated to distribute millions of seeds annually. So just 400 bats, millions of seeds, and this is a very common species in Latin America. And then bringing it a little more closer to the services that bats provide here in the Pennsylvania and Great Lakes region, mostly involved in controlling insect pests. So all of our bat species in the Northeast United States are insectivores. And I'm not a mathematician here, so I'm not going to get into all the number details, but if you break this down on a per bat basis, each individual big brown bat, which was the species they were studying in this report, can control insect populations by 250,000 every year. So one big brown bat, 250,000 insects controlled. So when we're talking about agricultural productivity and some of the points that were mentioned in the last presentation, This is one of the most common bat species we have in North America, so they are doing the lion's share of economic and agricultural services that bats provide. All right. And so we know how important bats are, and we obviously know how cute they are, so we can talk about maybe some of the conservation issues that are being presented to bats, particularly in our region. So around the world, bats have a terrible PR problem, so they have huge impacts from human persecution. I get calls multiple times a year of people trying to get rid of bats in their homes, and throughout the world, their entire colonies become exterminated, particularly in light of the COVID-19 pandemic. A couple other issues that bats face are the similar issues that most life faces on our planet right now when we're looking at climate change and pollution and habitat degradation and loss and things like that. And then in our region, bats are most impacted by white nose syndrome and wind energy development. And so the last presenters spent a good time talking about white nose syndrome, so I'll just kind of gloss over it a little bit. But it's a serious conservation issue that has led to 90 to 99% declines of some of our most common species of bat. So the little brown bat that was mentioned in the last presentation, one of the most common mammals in North America prior to white nose syndrome, and now they're very, very difficult to find. Although luckily there is evidence of resistance to white nose syndrome. They referenced a report from Michigan, and I'm also aware of a colony in the southwest portion of Chautauqua County, just north of here across the PA line in New York, where I've been monitoring a little brown bat colony for several years, and it does appear to be growing, so there is some really good news coming out of that. And then their graphic was much better than mine. They had the whole thing. I just have the current state of white nose syndrome, which has crossed both sides of the United States, going down into Texas. Although it should be noted that the records that you have for Texas are not necessarily white nose syndrome. It's more for presence of the Pseudogymnoascus fungus. So the presence of the fungus doesn't necessarily mean the bats have the disease, but the fungus is there to potentially cause that disease. And then moving on to talk about wind energy. So wind energy development is increasing every year in the United States and around the world as we try to get off of our addiction to fossil fuels. And unfortunately, it comes at a heavy cost to bats, bat species, particularly the migratory bat species. So in the United States alone, hundreds of thousands of bats are killed every year from direct collision with turbine blades or through a phenomena called barotrauma, which is essentially the small capillaries and blood vessels in the brains and lungs become ruptured by the differential vortices that are created from the spinning turbine blades. So this conservation issue is just as bad as white nose syndrome can be. They had mentioned in the prior lecture that about 6.7 million bats have been killed from white nose syndrome, and if you do the math, over the last 10 or 15 years, and you have hundreds of thousands of bats killed every year from wind energy development, you can quickly see that this is just as serious a conservation issue. In fact, recent modeling efforts have pointed to serious declines, particularly for migratory bat species like the hoary bat that you see here, which I don't want to get lost in a lot of the text on the screen, but just pointing out in this little red box, if you could read that in the back, this study modeled that hoary bat populations could decline by as much as 90% in coming decades. And in a more recent study, the modeling efforts suggested that hoary bats would experience a 50% decline by 2028, so just a few years from now. And they also, as you note on the bottom of the screen, that the current fatality rate of hoary bats is not sustainable and can pose a risk to the population, and that this turbine-related mortality may already be high enough to have caused substantial decline. So one of the basis for why I'm doing the research I'm doing, so I'm trying to address bat conservation issues related to white nose syndrome and wind energy development, and doing that through understanding the way bats interact with their habitat and a more thorough kind of assessment of that interaction. And I do this through collecting data on bat activity through ultrasonic surveys, where I go out with a special microphone and record bat echolocation calls and then analyze the data that I get. And so my hypotheses involve two things. So bat activity will be influenced in terms of habitat use by the morphology of the bat. So a big fat bat like the hoary bat with a big wingspan isn't going to want to fly and try to navigate through a cluttered forest. Whereas a small little bat like the northern long-eared bat, which specializes in flying in forests, would probably be more likely found in a cluttered environment. And then relative habitat value would be interacting too. So agricultural landscapes that have a heavy use of pesticides and chemicals might not have healthy insect populations, and so might not be the best habitat for a foraging bat species. So just for example. And so moving on to talk a little bit about my methods. So again, I do bioacoustic bat surveys. So I'm going out and recording bat echolocation calls. And bats use echolocation to forage and navigate. So they're emitting these extremely loud, high-frequency vocalizations and interpreting the echoes that come back to them. And by doing this, they can locate obstacles and fly around things. They can find food and insects that are flying in the night sky, or insects that are actually hanging on tree branches or on the ground or something like that. And they can actually tell how fast an insect is flying and the size of the insect. So they can discriminate between species of insect just using echolocation in pitch black. So very cool abilities that they have. And the echolocation calls of bats have 3 different phases: the search phase, approach phase, and terminal phase. And the search phase is almost like you're driving around at night flashing your high beams at kind of a standard rate. So the search phase of the bat's echolocation call is the one that you can use to identify species of bats. The approach and terminal phase get to be a little too variable because they're getting close to an insect and they're about to latch onto the insect in the approach phase. And the terminal phase is really, really rapid echolocation, thousands or hundreds of thousands of times a second, as they approach an insect and grab it. It's like a bzzz little sound to it. And so by kind of separating out the different calls that I record and looking at only the search phase calls, I can, for the most part, see what species of bat I am recording. And so as a response to wind energy development and trying to track populations of migratory bat species, the New York State Department of Environmental Conservation began a statewide population monitoring survey effort in 2009. So they would have volunteers driving around county by county, a 20-mile route, driving at about 20 miles an hour, so you match the bat's flying speed. And so this allows you to, you have assurance that each individual call you record is a separate individual bat. Otherwise, if you have a microphone just sticking up in the landscape, you have no way of knowing whether it's one bat circling twice or 2 bats circling once. So using this mobile acoustic protocol, you can get population level data, and that's what the state's been using that survey data for. This is what the microphone looks like, so it's not really as high tech as you might think, encased in a big PVC tube with a magnet that you put on the roof of your car. And they've been using the same microphone since 2009, so some of them are getting a little rough. And so I've been using the state's data and survey routes for many, many years. I've been working on the survey since, I think, 2013. And the one that's closest to my house is called the Jamestown Transect, which is shown here. So you have the black line running through the map that you see of the Cassadaga Creek watershed, and the transect itself runs from Bear Lake, New York, down to Falconer, New York. So this is the study area that I'll be using for the analysis that we did. The Cassadaga Creek watershed, which is shown in the delineation that you see there, and the survey route. The colors on the map are land cover. So you can see dark blue would mean open water, the lighter blue would be wetlands, the browns and yellows would be agricultural areas, and then the light and dark green would be forests. And you can also see there's not a lot of developed land in this watershed, but you can see a little bit of the city of Jamestown and Falconer right down around here where they enters the Chattacoin River. And so I go out and do these bioacoustic surveys and record bat echolocation calls, and then using specialized software, you can actually visualize the call. So what we're seeing here is a spectrogram, which is plotting frequency versus time. And by looking at the different spectrograms of each bat echolocation call, you can identify species. So I don't want to take the time to describe every little detail in the species identification process, but the table on the left is a result of some of the initial research I published back in 2021. So these are the parameters that I use to identify the bat species from these surveys. It isn't without complications, so not all bats produce species-specific vocalizations. So the big brown bat and silver-haired bat are actually quite similar in terms of their echolocation call. And you really need a couple specific things to be able to identify these species accurately with these methods. So on the left, we have the big brown bat, and that spectrogram can look very similar for big brown or silver-haired bats, except when you have, and this gets a little specific, you have an initial frequency starting above 60 kilohertz. Then you know that's a big brown bat. Silver-haired bat calls, to be the diagnostic call, so to speak, that you would get, would be one that's mostly flat, ending at about 25 kilohertz. And so taking the data that I collect and analyzing it to species, we can then do some statistics on it. So I use what's called logistic regression modeling, which is a statistical technique that can provide predictive models. It requires absence as well as presence data. And a lot of scientists use it and biogeographers use it to create maps of habitat suitability or foraging suitability. With logistic regression, I was able to compare bat echolocation activity with land cover values, and we did this using the presence data that I recorded, as well as pseudo-absence points. So using coding, we were able to extract pseudo-absence points from the map, and so these were points on the map that were not a bat, and were at least 60 meters away from any other pseudo-absence point. And so that gave me some statistical outputs to compare bat calling behavior with discrete land cover variables, so we could see which land cover variables were most important. We were also able to take that information and process it again to create habitat suitability maps. And we did that through use of the logit function, which you see at the bottom of the screen, which is essentially taking bat presence information and the probability of recording a bat within 30 meters of a given land cover type. The third kind of major chunk of this paper was looking at echolocation density analysis. So in ArcMap GIS, you can do a point density analysis. So we're looking at how many echolocation calls were recorded per a given unit area. And then I was able to relativize them with average, above average, and far above average echolocation call density, which is what you're seeing on the map here. You can see these little yellow blobs, which would be average, orange blobs, which would be above average, and the red blobs would be far above average. So I was able to go and find areas in the Casanova Creek watershed along this survey route that were above average echolocation calling density, which means that there might be some way to use this information as sort of a proxy for some environmental variable, or maybe there's a colony nearby, so you just have some different density in bat calling behavior for some reason at these points. So moving on to the results, I had data from 2009 to 2020, so I had 7 years of data. Not every year had data for it. We were able to extract 1,600 occurrence records from 5 different species of bat. So they're listed down below, about 560 big brown, 560 silver-haired, 143 eastern red, 260 hoary, and 78 little brown bat calls. And I went through the land cover data and aggregated the land cover variables into just 5 basic types. So we had open water, forests, which include coniferous as well as deciduous forest types, wetlands, which included emergent as well as, something like a red maple swamp, different agricultural areas, and different developed areas. And we found some significant interactions. So this is the first output from the logistic regression analysis. So you can see most of the species had a significant interaction with forested habitats, a significant positive interaction, so those would be shown in green. You also see a lot of significant negative interactions with agricultural habitats. So that's kind of lending support to my hypothesis that agricultural areas may not be the best foraging habitat for certain bat species. I also found bat echolocation hotspots that appear to be fairly species specific. So the only two that overlap significantly would be the big brown and silver-haired bat. And so that's why I made a sixth map that shows those two phonotypes. So that's kind of the phonics of the echolocation calls lumped together, because remember, there are some species identification issues that are kind of associated with those two species, as I mentioned a few minutes ago. And then I was also able to create habitat suitability maps. So here we're looking at the output from that logit function analysis and kind of taking the different probability of recording a bat species and putting that through the lens of land cover. And so here we have bat habitat suitability from least suitable, below average, average, above average, and most suitable. And so just a few minutes to bring this all together and discuss this kind of species by species really quickly. So you can see the big brown bat habitat suitability map shown here on the left and the echolocation hotspot on the right. And if I just toggle through, we have now looking at the silver-haired bat. They do look very similar. However, looking at the large area in the center of the Cassadaga Creek watershed here, you note that the big brown bat has lower habitat suitability in those cluttered wetlands than the silver-haired bat does. And that is really interesting because there's issues with discriminating between these two species, but this process still was able to show some information that also aligned with what we know of the natural history of these species, which is that the silver-haired bat does tend to forage in wetlands and big brown bats do not. So it's very interesting. Moving on, we have the eastern red bat and hoary bat, which are both migratory foliage roosting species, and they showed very similar habitat suitability. However, their echolocation hotspots were at very different points on the map, so there might be something more that we can learn from that analysis there. And then moving on to talk about the little brown bat, which has been so hard hit by white nose syndrome. Not a lot of habitat suitability, so if you're looking on the left, but what we do see of the dark blue, most suitable or above average suitability, is right around places like Cassadaga Lakes and Bear Lakes, which, as we know with little brown bats, they tend to forage over open waters and tend to live near water bodies. So this analysis that I'm doing is aligning nicely with what we know of the natural history of these species. And just a few minutes to talk about, not even a few minutes, a few seconds, I saw the sign. A few seconds to talk about the next steps. In 2022, I expanded my survey areas. So we have in black the Jamestown transect, which was part of that long-term analysis that I've been discussing. But we also have in yellow the Ball Hill transect, a Lake Erie transect, and a Niobe transect. So I added 3 more survey routes for the mobile acoustics, and I've got several different sites for the point survey locations. So I'm going to expand the analysis I did, add in a few different more environmental variables, and do this analysis at the Huck 10 watershed level. So it would stretch from Cattaraugus Creek in the northern parts of Chautauqua County all the way down to here in Erie, because we're all part of the same large-scale watershed system. And then these are some of the different environmental variables I would be plugging into the model and see what we can come out with for more of not habitat suitability, but more species distribution modeling. And those are my references. I promise they're real. You can ask me and print them out for you later. If I have any time, I'll take some questions now, or you can.
Actually, if you wouldn't mind waiting until our break.
I have no problem with that.
Yeah, thank you so much. Fascinating.
Thank you.
And let's see what happened to this thing. Are you sure you don't mind? Okay. Okay, our next speakers are Jewel Gonzalez and Macy Heptich, and they are presenting a study of bird building collisions during migration seasons in the city of Erie.
Good afternoon, everyone. My name is Macy Heptich. This is Jewel Gonzalez. We worked with Dr. Stephen Robsky and Dr. Sarah Sargent in a study of bird and building collisions during migration season in the city of Erie. So our purpose during this research was to monitor the hotspots in downtown Erie for birds that have collided with windows during their migration. So there are a couple reasons why this happens. We're going to start off with the daytime, and in the daytime, the reflectivity and transparency of the glass actually makes birds really hard to see through the glass, and sometimes when there's a path to greenery and potted plants, the birds will actually try to fly to those plants to land on them. In addition, during seasons where territoriality is high, such as mating season, birds sometimes look at their reflections in the windows and attack it to assert their dominance. In addition to daytime, we also looked at the nighttime, and as you can see at the bayfront with the Bicentennial Tower, highlighted in the middle here, the lights and exterior lighting can be very disorienting for birds, and this is also very prominent when there are certain weather conditions such as fog, snow, and rain. So we worked with the Erie Bird Observatory, and they are dedicated to fostering enthusiasm for birds and conservation through ornithological research and public engagement. They offer many different programs highlighted on the slide here. And on the right side, we have all of their contact information if you are interested in becoming involved with them. So within the Erie Bird Observatory, there is a program called Bird Safe Erie, and Bird Safe Erie is aimed at protecting migratory birds that travel along the Lake Erie shoreline, which is a vital path for migrating birds. There is an important rest stop at Gull Point on Presque within their migratory path when they are coming from Canada and then flying south. Bird Safe Erie also monitors and identifies ways to reduce the risks of light pollution and urban infrastructure for these important visitors. So during migration season, collisions with buildings are a major cause of death for these birds. The Erie Bird Observatory is studying this in the city of Erie, Pennsylvania, in both the spring months of April and May, as well as the fall months of September and October. So it is also estimated that approximately 988 million birds die every year in North America due to colliding with glass, including windows and buildings. To put this in a larger perspective, other than in Erie, Pennsylvania, in Chicago on Thursday, October 5th, approximately 1,000 birds died after striking the windows of the McCormick Place Convention Center. And during that day, there was over 100,000 birds migrating across Chicago. And there's recent evidence that has pinpointed the climate change impact on birds. And this is the most they've collected within 40 years of migration.
Now onto the physical aspect of the research itself. Here you can see the little stars representing where each specimen or deceased bird was found. Also in red, you can see that there were 2 routes taken both by faculty and students at Gannett University, each in the morning, roughly between 6:30 and 7:30 in the morning. When the bird was found, the location, time, and the species of the bird were then recorded. Injured birds, if found, were taken to the Tamarack Wildlife Rehabilitation Center, and then dead birds were frozen for then future identification. The goal for this research was to identify which buildings had the most incidents, and then to figure out how we would lessen these through modifications. I'd also like to make a note that there was also 4 other routes in the city where identical work was being done with the Erie Bird Observatory. If you're from Erie, you can kind of orient yourself. Right here would be Gannett's campus Nash Library, right off of West 7th and then off of West 6th. Right here would be the courthouse. Right here is State Street where the Avalon Hotel is. You can also see over here would be our Zern Science Center, Peach Street, whatnot. This would be one of the bags that we would place the specimen in. Would record things such as the date and time of collection, as well as what side of the building that the bird was found on in comparison to the lake. Yep. Onto like the bird handling and safety aspect. Gloves were provided within the bag. I'd also like to point out we would wear one of these tags to walk around with, so people weren't like, why are these kids picking up dead birds? So we get to show them our nice little tag, as well as hand out one of these brochures to see if they wanted to help out. If we did find a deceased bird, we would take them and place them in one of our bags and then fold it. Injured birds were then also placed in a bag, first put under the net that we would use, which is also right here, brought this with us. And then we would then message Dr. Sarah Sargent and inform her of the injured bird. We also would use an app within our cellular device to help with the figure out where the bird was found. This is the data that we were able to collect. On the left-hand side, we have where the building that the bird was located at and their date. And then on the right, you can see the number per species of birds that were found. All birds that were found, I'd like to note, were deceased. We didn't have a severe trend since this is only the first year that we've done this for species of bird. I'd like to point out the American woodcock that was recently found. I'd like to point out its preferred habitat to be the young moist forest that was found on campus, so I thought that was super interesting. As well as down here, the last 4 birds that were picked up were the night of that snowstorm. So I'd kind of like to, if you could kind of think of driving through the night with a serious amount of snow coming down your face. I mean, that'd be pretty disorienting. Now onto the buildings themselves. Our biggest culprit was the north side and south side of our Nash Library. As you can see on the north side, those windows are ginormous and pretty reflective with the transparency too. Same thing with the south side. This was our biggest culprit for the birds found on campus. Next we go to our Zern Science building, also off of campus. In the picture to the left, you can see on the right side we do have classrooms with some transparent and reflective windows. However, once we discussed with Dr. Ropsky, we had agreed that height was probably a factor for this building. Next, this one's kind of just a doozy. We weren't really sure as to why we found a bird in front of this building. This building is just right off of West 8th Street. We actually believe it could have been, it could have happened like due to another infrastructure off of maybe Sassafras. There's like a retirement building. We think that have collided, and then the elements with wind and stuff could have brought the bird then in front of this building. Our next building would be north of Cathedral Preparatory School. But again, actually, we weren't too sure as to why we found this species here. But once we looked to the left-hand side of this picture, you can notice St. Peter's Cathedral. And again, if you're from Erie, that building is lit up all through the night. So we believe the bird may have actually collided into that building and made its way in front of Cathedral Preparatory School. This one, this bird was found in front of the Knights Club off of campus. Again, not really too high, not too transparent or reflective. So we looked a little further down the street and we noticed one of our freshman dormitory buildings has this pretty big reflective and transparent window on the west side. So again, this could have been one of those situations where the bird had collided and made its way down the street. The last one would be the specimen found out front of Rite Aid. Again, not really weren't too sure initially, but once we looked across the street, there's this 13-story building that stands pretty high compared to the rest of the infrastructure in downtown Erie. So we believe it could have collided with this building and then wind taking it across the street. Now to the important part. There are ways to minimize the bird deaths, and just these are some of the strategies. You can make your windows obvious to avoid confusing the birds. You can not use landscape lighting to light up trees or gardens where birds might be resting. You might also close your blinds at night to reduce the amount of light being emitted from windows. You can add interior and vertical blinds and keep the slats half open. And you can avoid visual paths to sky and greenery, such as interior plants near the windows. And then our goal on campus from now on would be, since we've identified Nash Library as the biggest culprit on campus, we will be reaching out to the director of Nash Library to see what adjustments can be made to minimize this effect. We also plan to continue the partnership with Dr. Sarah Sargent and the Erie Bird Observatory to continue this research. We also want to thank the people listed below for taking time out of their day to go on these walks, regardless of weather, rain, wind. So yeah. Thank you. Any questions? Holy crap. Okay, go ahead. Here, go ahead.
With the library, do you have a gut feeling on whether it is nighttime illumination or the reflectivity of the windows that might be more the culprit?
Yeah, so I actually work at Nash Library. I'm a supervisor, and I am also aware that the hours of illumination run from about 6 in the morning, and they're on till about 12 at night. So I really think it could be a factor of both of those things. So again, we are talking to the director to maybe manually be turning on these lights and turning them off instead of them automatically be turning on. Whatnot. Yes.
Was part of your surveying path adjacent to the new cybersecurity building? Because that surprises me that that's such a big, bright window building that you didn't find any there, or was it just you didn't survey that area?
Many of us have actually extended the survey route, so there were times that we did make our way out around there. Again, there weren't any found in that area. And again, it could have just been the times that we were picking up the specimens that other animals had gotten to them first, picked them up, or they were just disoriented and injured and had made their way off, flying away. So, go ahead.
Can you go back to the slide about reflectivity? Okay, cool. Is there, I don't expect you to know this, but is there a measure to measure reflectivity and transparency, which are opposing things, or opposing qualities of windows? Is there a way to measure that?
I mean, a measure of transparency could probably, this is a very surface level answer in a way that how much you can actually see through that, and if potentially tinting a window would make it better, but then again, that reflectivity would probably counteract how transparent a window is. If you tint it more, then you increase the reflectivity. It kind of, so.
Yeah, because if you're looking at a window head on, it's likely more transparent into the view, and then if it's reflective, you're looking at an angle. When you know the measure itself, you can also dictate, okay, if you're at a 30-degree angle from the window, not directly perpendicular, it's more reflective that way. And that just goes back to another thing you can propose for the solution.
Yeah.
And it's that these windows, you can answer this in okay, based on the trends of your buildings, these reflective buildings, are they north-south to the parallel with the migration of the birds? Because the angle of the building where most of the windows are, you can probably glance it. So if the birds are going true north, would it help if those buildings, if all of them aren't also true north, but they're 40 degrees to the migration? and instead of perpendicular or like 30 degrees?
Yeah, that's an excellent point for sure. It's a bit difficult to say if they are migrating from true north, just because depending on where you're looking, if we're facing the building, the north would be, I guess Canada would be on the west side coming across, since the way we face the bayfront. But yeah, that's definitely an excellent point.
Okay, so we talked about the window quality, we talked about the physical alignment. What about the geometry of the windows? Is there, I don't know, like concavity shapes?
Right. Granted, not too sure if that would help, just because depending on if they're concave, convex, or just completely a straight window, I'm not too sure if that would help, just because you still would have that reflectivity as well as the potential for transparency and that path to potted plants on the inside of the window. So, yeah.
Okay, so if we're looking at, sorry, I might take too much time.
We have like 1 minute.
Oh, okay. So, okay, we're talking about like, you know, how clear the view is for the birds. But these geometries as well. If a bird hits like, maybe like a convex mirror versus concave mirror, can it also like reduce the blow?
I guess it, once again, will come back to the angle at which they hit that, because if they're flying hard and like fast enough and are already disoriented, I'm not too sure if that would help reduce the blow, because if you still run into a convex or a concave like mirror or even a window, depending on how fast you're going, it's still gonna hurt and still gonna potentially break their necks. So we've had a couple birds with broken necks, so yeah.
Just some things
Yeah.
I want to consider.
Okay, thank you very much.
Thank you.
There we go. So are you all right with just advancing on the, are you all right with just advancing your slides on the computer?
Yeah,
You
that's
can
fine.
still use the laser pointer on that. Okay, our next speaker is Holden Cooper, and he's presenting There She Glows, cover, Patterns of Biofluorescence in Snakes.
Hi everyone, so my name is Holden Cooper and I'm presenting my research, There She Glows, which is patterns of biofluorescence in snakes. So to start off, what is biofluorescence? Biofluorescence is where a high energy wavelength of light is absorbed by an organism, then re-emitted at a lower energy wavelength. And so a good example of that is seen in this chameleon here, where the chameleon is exposed to UV wavelengths of light, and then as a result, biofluoresces blue, which are these spots right here, and this is biofluorescing through the bone. So this is something that differs from bioluminescence, in where bioluminescence is where the organism produces its own source of light. And in this example with the deep sea shrimp, the shrimp is kind of expelling bioluminescent goo, basically, which is a way to disorient a potential predator, especially in this deep sea habitat where it'll just kind of stun the predator and allow it to make an escape. So overall, we don't really understand the ecological significance of biofluorescence. However, we do have some examples where we have some more explanations. And so we can see that in flowers. And in this example here, we have a flower where the nectaries are kind of biofluorescing in response to UV light. And these nectaries allow pollinating insects such as bees to kind of, they act as guides, guiding the bees into where the nectar is on the plant. Bees tend have ocular sensitivities to UV light, so they're able to pick up on these specific biofluorescent signals. So biofluorescence is pretty well documented in a lot of marine systems. However, we're only just beginning to uncover biofluorescence in more terrestrial systems. And of these more recent studies, they tend not to include spectral data. And spectral data is very necessary because this is a more concrete way to quantify the colors that we see biofluoresced. And so here I kind of have visible light and then UV light at the end here and then infrared here, but different wavelengths kind of just represent the different colors that we see. So having spectral data is just a concrete way to kind of confirm that this is the color that we're seeing biofluoresced. Additionally, very few studies documenting biofluorescence in terrestrial systems actually focus on snakes. And so to my knowledge, I was only able to find 5 sources which document biofluorescence in snakes. The most comprehensive of these I've put here at the bottom, which uncovered biofluorescence in response to UV light in many viper species. So I had 2 kind of objectives with my project. The first of these being, is biofluorescence present in snakes? And the second, what variation may lie between different species? And so for these next couple slides, I'm kind of going to keep it with a couple of species on per slide, as these can tend to have variable morphology. I'm just going to briefly explain that. So to start off, we have the gray rat snake, and at its younger juvenile stages, they tend to have more pale coloration with dark splotches, and then adults will have this dark marking on the dorsum, and ventrally they tend to have more lighter colors, especially in that upper portion of the ventral surface. For milk snakes, we kind of see 2 different morphs for milk snakes, where they can have this kind of brown patterning here, or they can be more red colored on their patterns. Northern water snakes, the juveniles, tend to look quite similar to rat snakes, and they kind of have the same kind of lightly colored in the between marks, but then on marks it's much darker. As they age, they become darker in color, and you see like more dark brown to light brown in between. Northern red belly snakes, the main morphs that we really see are this slate gray one and the reddish brown one. And as the name suggests, they have a red belly. And the purpose of this is to act as kind of warning coloration for a potential predator. The remaining species don't really have any variable morphology. So we have the decayed brown snake, eastern garter snake, short-head garter snake, and the ringneck snake. Ringneck snakes, much like the red bellies, have this kind of bright warning color on their bellies, which they use to kind of frighten predators. And so here I've kind of mapped all these different species onto a cladogram, kind of showing the relatedness, and this will come in more useful as I discuss the results. So for our study sites, I've used my research advisor's faces against their will to mark where we sampled snakes. And then just for those unfamiliar with the county, I've used my displeasing face to mark where we are currently, hopefully to help us better remember where we are while walking around a study site until we see a snake. Second of these, we would also lay out cover objects, such as cover boards, and these would generally consist of wood or shingles. Leave them out for a period of about 2 weeks, kind of giving it ample time for it to be more preferential for snakes to go underneath. Flip these, and ideally we'd find a snake underneath. So when we do find snakes, what we would do to document their pattern is first, we would take photos of the dorsal surface, then ventral. Then we would expose them to either UV, blue, or green light. So we tested these 3 different light sources to determine like what biofluorescence responses they might have to these varying light sources. And then we would use a filter to block out any excess light from the actual light source. So the snakes will kind of absorb the blue, for instance, and they'll absorb blue light, then re-emit that as green. However, there's still going to be some background blue light, which would get into the camera and kind of drown out what we're seeing biofluoresced. So we have corresponding filters for each wavelength of light that kind of show us, block out the wavelengths of light that are being tested, basically. So for here we have UV, and this is a 450 nanometer filter, meaning that any wavelength of light below 450 nanometers is not going to get through the camera. So this is going to ensure that we're only seeing what specifically is being biofluoresced, and allows us to kind of document what that pattern is. For collecting spectral data, we would use very similar methods. However, we would be using a spectrometer, putting this on the dorsal or ventral surface, and collecting data on what colors are being biofluoresced. And again, using the corresponding filters, so 415 for UV, 500 for blue, and 600 for green. So, did they fluoresce? What we found is that these snakes biofluoresced at all wavelengths of light. However, in response to green light, they had the most minimal signs of fluorescence. And so, going just from left to right, we have an example of UV fluorescence. This is blue light fluorescence, so typically we saw a lot of green as a response to blue light. And then here we see green light response, which is more red. And as you can see, it's very minimal. However, a pattern still is present here in between skips. So just to kind of generalize the 3 patterns that I really saw, We have this first one where the snakes were biofluorescent in between their kind of dorsal markings, been mildly fluorescent on these markings, and I include a photo of that here. Another one where they have actually multiple colors of fluorescence in response to light, and we would see this across UV and blue light. So in this example, we have a garter snake biofluorescing both green and orange. And then this last pattern where biofluorescence is very dim and almost not noticeable. So, and we have that with the rat snake here. For ventral surfaces, we saw two main patterns, where the first of these being all kind of underside scales would biofluoresce except for their labial scales, with the labial scales just being chin scales. The second pattern we would see ventrally is just all scales lighting up in between. And so going back to that cladogram I had mentioned earlier, here we just kind of have the different patterns of fluorescence dorsally that are just kind of the main ones that I saw. So to start off, I just marked this pattern with this kind of spotted line, and we only really mainly see that for the eastern milk snake, where in between their spots, they're quite intensely fluorescent, and then on top of the dorsal markings, they have some minimal fluorescence. The remaining species all had this last pattern where it was very mild and almost not noticeable, and this was in response to all UV, green, and blue lights. For ventral surfaces, we do see a bit more variety. So the eastern milk snake and the gray rat snake in the family Colubrinae here would display this first pattern of fluorescence where labial scales would not light up in between the scales. The remaining groups, remaining 5 species in the family Natricinae, so this would be the water snake, brown snake, red belly, and both species of garter snake, all displayed this last pattern of fluorescence here, where in between scales, they would have the most intense fluorescence. We also saw some variation in fluorescence within the species. So to start off with the milk snake, we would, in one individual, which was that red morph, the only red morph we were able to find, it actually displayed red fluorescence in response to blue light. So it'd be more of this pattern here. For the rat snakes, since they have variable appearances in the juvenile and adult stages, we saw that juveniles were actually more of this first pattern where they have fluorescence in between these different markings, and then also have fluorescence on the actual markings themselves, just minimally. The same was true for northern water snakes, which have a very similar pattern of fluorescence and just similar pattern overall. Brown snakes were largely the same, and red belly snakes and eastern garters and the ringneck snakes all displayed multiple colors of fluorescence, with the ringneck having the orange belly at both UV and blue, and then the red belly snake having fluorescence at UV and blue as well. We didn't really notice much of the color through the green light responses. So here's kind of just the spectral data we collected, where I have the count representing intensity of light and the wavelengths at the bottom, and then each line is just a different color line is representing a different species. And an important thing to note with these graphs is that we don't really want to focus on the intensity of light, since flashlights were held at variable distances. This isn't an accurate reading as to how intense the light was. The point of this is more to look at the pattern, where we see, in response to UV dorsally, all species kind of exhibit fluorescence at this wavelength here, which is close to teal. When we look at ventral surfaces, overall, it is more intense. However, once again, we don't really want to compare that, as it's not a very accurate measurement. However, from what I saw visually, we see that fluorescence is more in this kind of greenish teal, and is most fluorescent there. So for different species, what I just saw visually, however, we would see that like rat snakes and eastern garters were more minimally fluorescent, and then short-headed garter snakes were actually some of the more fluorescent species that I saw, which isn't really well represented here. But for blue light, we kind of see this similar trend where snakes are all displaying that similar pattern of fluorescence around the same area, and in this case, it would be green dorsally. And for those species I had previously mentioned with variable colors of fluorescence, we would kind of see, like, for instance, this purple line representing the red belly snake. We see some red over here, reddish orange, and the same thing being true for the eastern garter, where there's a little bit more of this orange here. So that just kind of shows that they have more variable coloration. And this is especially more pronounced here, where, especially on that ventral surface, we really see the more vibrant color for since some of these orange colored or different colored markings are located more ventrally than dorsally. So green light is more of the exception to looking at the actual counts of intensity, where green light responses were so minimal visually and through the spectrometer, it was very hard to pick up anything. However, there's still a pattern present, where typically around this red we see fluorescence in all individuals, although it's just very dim and hard to see. Ventrally, we see a little bit more noticeable fluorescence, and it's a little bit more of this deeper red, but overall, it's just very minimal and likely not all that significant. So, just in conclusion, ventral surfaces were the most vibrantly fluorescent regions, with green light resulting in the least intense fluorescent responses to light, and some species had additional colors of biofluorescence present. So, important question to ask is, why should we care about this? And the more we know. So, knowing more about what biofluorescence is a very novel field, we're only just uncovering just that species are even capable of biofluorescing. So we don't really understand why it's significant. So these studies that are documenting the presence of the trait are important as they kind of highlight, or they will allow us to kind of conduct further study into this. And some ideas for why this might be is it could be used for counter shading. And so snakes will engage in this behavior called periscoping, which we can see in this racer here, where they lift up the upper portion of their body and will kind of scan their surroundings. Doing so is going to make themselves more vulnerable. And considering that ventral surfaces were among the more vibrant areas that we saw fluoresce, this could be used to kind of help them camouflage better. So since a lot of the species documented that I found inhabit more open areas, having fluorescence on the underside while periscoping would be incredibly helpful for blending in plants that may also biofluoresce. Additionally, aposematism, it could play a role in aposematism. As we see here with this red belly snake, there's some red fluorescence on the underside. It could further enhance biofluorescence, or biofluorescence could further enhance these warning signals and make it more difficult or more threatening for a predator. So I'd like to thank Penn State Barron for funding this research, Mark Letheby for aiding in data collection, Ripley Kindervarter and Sophie Bond for data collection, and Beth Lass for calibrating our spectrometer. Does anyone have any questions? Yeah.
Can you use historical images of snakes to do something like this, or do you have to actually be shining with the UV or other?
Sorry, could you say that again? I couldn't quite hear it.
Can you use other images of snakes, like historical images or photographs that other people have taken to do research like this, or is this something you have to catch the snake?
So we would need to physically have the snake. So, because what we're looking at is a response to a different wavelength of light. So we're shining like a blue light on it and putting this filter over it, just showing what pattern they have as a response to this. So we can't really test that from other photos. We need a live animal for that.
It's really interesting that the curves remind me of a remote sensing we use to discriminate from bare rock and soil, plants, and water. So it's kind of a really interesting thing.
Yeah, thank you. Yeah.
Do you have permits to handle the snakes at all?
Yeah.
Was there anything done to ensure that there was no fluorescent fungi or spores or bacteria on the ventral side? Because they're going to be rubbing against stuff and it might be just picking up and interfering with the natural fluorescence.
Sure. So I guess to kind of account for that more, we would sample multiple individuals, and these are found in variable habitats. And overall, we would just kind of see the same patterns of fluorescence within individuals. There wasn't too much variation, so I think that would kind of account for that.
Well, that's all the questions. Thank you very much. You ready?
Born ready.
Okay. All our stuff's broken now. Come on there. Was that slide from here. All right, our last speaker of this session is Sam Wilczynski, and he is presenting on the impact of sexual cannibalism on male fitness in Tenodora sinensis, or the praying mantis.
All right, so as she said, I'm going to be assessing praying mantis, Tenodora sinensis' fitness in the males in relation to sexual cannibalism in the species. Before that, a little bit about myself. I grew up here in Erie, PA. I got my undergrad here at Gannon, so I'm true bred local. And I'm now studying at SUNY Fredonia, pursuing my master's in biology with hopes of getting into wildlife biology. And that's actually a picture of when I worked here at the RSC. So that was a couple years ago. So I've been lucky enough to work on this park for a couple years before. So what is Tenodora sinensis? Tenodora sinensis is a species of praying mantis, so the order Mantidae. It's also known, the common name is Chinese mantid, so it is from Asia originally, but it was introduced here in the United States around 1896. It was introduced as pest management in an herbarium down by Philadelphia, and it didn't do a very good job of relieving the pests there, and it just escaped the herbarium, and now it is basically found all across the United States. The conservation status of it is mixed depending where you find it. Some states will dictate it as an invasive species, causing problems for other native praying mantids. Here, not so much. There's really only two species we find here, Tenodora sinensis and one of its close relatives, Tenodora augustipensis. So this is just a picture of what it looks like. Males are going to typically be way smaller than females. The way you can tell Tenodora sinensis is the brown backing and the vibrant green abdomen. There's also some things you can look at on the head plate. There's like basically you either have a triangular head plate or a square head plate, and you're looking for the triangular one. And then on the left there, that is the egg or the ootheca, which is basically what we used. We collected these in order to learn more about the mantids, sexual cannibalism, and mating. So why should we care? Obviously because of this amazing behavior that we see in this praying mantid. It also occurs in many other species of praying mantids. And this is sexual cannibalism. So if you don't know what that is, it is simply the female will either consume the male, with this unfortunate fella, during, before, or after copulation. And interestingly enough, even if he's eaten, he will finish copulating with the female. So will have successful sperm transfer, even if his head is completely removed. So sexual cannibalism occurs in Tenodora sinensis about 14% of the time in the wild. That's about in the middle ground of other species, so it's not like hyper common, but it's also not really rare. So, and then in sexual cannibalism, we actually see there's been research showing that there's kind of two different forms of sexual cannibalism that have evolved. We have self-sacrificing rituals or behaviors, which we can see in this fishing spider here. So this is the male on the back, and it's hooked on and started transferring sperm. And at the moment that its specialized genitalia attaches and sperm transfer completes, the male instantaneously dies, basically. It goes rigid into rigor mortis. That way the female can immediately consume it, and it contributes greatly to oogenesis or egg production. And they tracked the resources, so the proteins in this spider ended up in the eggs of the female. And then there's also involuntary cannibalism. That's what we see in Tenodora sinensis. So the males aren't willingly sacrificing themselves to the female to have better offspring chances, or their offspring to have better survivability. So I'm focused on the male fitness here, but there are benefits and losses for females and both. So there's this give and take. So the mating system of its closest relative, Tenodora augustipensis, is polyandrous, which is what we suspect Tenodora sinensis also is. And all that means is that females are mating multiple males. And then a not so close relative, the RSC, this is found over in Japan mostly, is actually a monogamous system. So even in the praying mantids, you still have these monogamous systems with no sexual cannibalism. And based on field observations done by my lab's predecessors, it's kind of hard to see, but this is the female. You can see that size difference. And then we see actually 2 males on top of the female here. So based on this and some of our field data, we believe that females are attracting multiple males, and those multiple males end up mating with that female. So they use sex pheromones. Tenodera sinensis uses sex pheromones to attract the males. So we see this a lot in the field. And then also I just want to talk about the sex ratio. So at the beginning of the mating season in the Chinese mantid, it's close to 1-to-1, so a 50-50 split. If you have 10 females, you're typically going to find 10 males around it. This slowly trends down as the season elongates, mainly because the males are the more mobile of the two. So females, once they become reproductively active, they kind of just sit down and stop moving and call in all the males. So the males are flying around, moving much further, 3 times as far as a female does in a day to try and secure mates. So that makes them easy prey for birds and other predators, as well as they just get eaten over time by the females. So what is our working hypothesis? If Tenodera sinensis exhibits polyandrous mating with a multiple paternity, so multiple males being represented in the offspring, then male fitness must be influenced directly by acts of sexual cannibalism. Because males may be missing out on additional mating opportunities, which will greatly reduce their offspring produced. I forgot to mention the ootheca. Within a single ootheca, you can have 40 to 200 nymphs. So if you even miss out on having a second ootheca or contributing to a second ootheca, you potentially are missing out on a lot of offspring, just tanking your fitness. So the main goals of our study: determine the mating system. We want to find the conservative estimates of male paternity, so how many males are contributing to a single ootheca, and we want to use that estimate to kind of generalize the natural mating frequencies, which will give us insights on male choice, male risk aversion, and ultimately answer how much is sexual cannibalism affecting male fitness. So how do we plan to do this? To do this, we are basically looking at the number of male mates a single female has, because a female can only produce, there's only one female producing the egg. It's not like 2 come together and make 1 mega egg. So that way we're able to determine, do females mate with multiple males? And we do this through microsatellite analysis. So the general methods are, we go out, collect them, we do some genetic extraction of the nymphs from the ootheca, and then we use this microsatellite analysis technique to determine how many males are in that ootheca genetically. So our sampling sites are just old growth fields. These are successional fields from abandoned farmland and such. So these are typically filled with like goldenrod or thistles, and these are just tall, late season blooming plants, which mantids really like, so that's why we picked those areas. We used a simple transect survey, just walking straight lines through the fields until we sampled to completion. We ended up having 4 sample sites, 2 of which were on SUNY Fredonia's campus, and then one is Canada Way Creek, which is just a creek that is adjacent to the campus that has many of these fields adjacent to it. And then the Fredonia Little League fields, so just a little bit of goldenrod here and there, big open space, a little bit of goldenrod here and there. And then we also had some students from Fredonia bring in ootheca. Unfortunately, we didn't get adequate geological locations for those, so in some of my other analyses that we have to remove those ones. So what are microsatellite markers? Microsatellite markers, or simple sequence repeats or short tandem repeats, you can use those interchangeably, are just repeating units of nucleotides, 1 to 6 base pairs long. And if we look here through the laws of segregation of alleles, these are all siblings on the right. So what we're looking at is that's one allele, that peak there, and that's a second allele. So those are microsatellite sites that are amplifying. Here we see sibling 1, sibling 2, and sibling 3 all share this allele, which is a homozygous mother. So they're receiving one copy of this allele from the mother and one copy from a father, a different father, and a different father. So that's why we use microsatellite markers. They're really highly polymorphic, which just means there's lots of different genotypes. And this is even used in humans for like paternity testing and like disease testing, like rare diseases and everything. So you can use it in all sorts of systems. It's great. So we at Fredonia, I didn't, some students prior to me created novel markers, which are just sites at which we're amplifying to get the gene there. We have SK1, CRAM8, and SDDS4. You can see, or you might not be able to see, these are in blue and this is in blue, but this one's in green. The only reason that matters is these two loci overlap in base pairs. So to be able to tell which peak is for which primer, we had to fluorescently dye them differently, but it doesn't change anything about how you read it. All you're looking for is the tallest peak, that being an allele, and that being an allele, or here we have two different alleles, so that tallest peak would count, and that tallest peak would count, and then we have two alleles here. So the process is pretty straightforward. You just extract DNA from the nymphs. You do a PCR with these novel primers to amplify at those sites. We used a special program called a touchdown program to stop some non-specific annealing. Then you put it in your gene analyzer and bam, you get your results. So what did we do with those results? We did a parentage analysis in a computer program called Jared 2.0. In order to use this program, you need a progeny array, which is just listing your offspring's genotypes out in a really annoying syntactic way. It's just a bunch of coding. And allele frequencies, so just the rates at which they appear in your population. And the program gives you the possible mother genotypes along with the minimum number of males that are required to explain your progeny array genetically. And then it also gives you different combinations of those possible father genotypes. So this is what the program looks like. So this is our progeny array here. These are just genotypes. So this is for SK1, CRAM8, and SDDS4, and the numbers are just the allele peaks, so, or the base pair at which we found that allele. And then on the right is just allelic frequencies. Then the next step is we need to determine the mother, so question mark, question mark, question mark up there. Based on this and the laws of segregation, it tries to find which allele is reoccurring at those sites. And in this example, there's only one possible explanation. Sometimes, depending on the progeny array, you might have two genotypes for the mother, and it just tells you which ones it could be. And then the next phase, which is our important phase, is it tells us how many fathers had to be represented in the ootheca conservatively to explain that progeny array. So this isn't actual mating frequencies, unfortunately. This is just the minimum number to explain it. It can't calculate, because then it would just be doing a bunch of assumptions, and it would probably take weeks to calculate and do a ranking. But this is what we want, so we know that 3 fathers are represented in that ootheca. So our results being, we found 22 ootheca, and we sequenced over 300 nymphs at those locations. And sequenced nymphs is just like, I coined that, nymphs that amplify all 3 loci, because in order to use this program, you can't have any null alleles in that progeny array, or else it'll just error out. So at all 3 sites, we get the most information. And our paternity results end up being 3 ootheca came back with a single father representing it. 12 came back with 2 fathers. 3 came back with 3 fathers, and 1 came back with 4 fathers. So 19 out of 22 worked properly in Jared, and then 16 out of 19 processed with multiple paternity or multiple fathers, so ranging from that 2 to 4. On average, it was 2. So what conclusions can be made? Well, first off, the novel primers work. They're effective and practical. I worked along with one individual sequencing these individuals over the summer, and we were able to sequence over like 600. They didn't all amplify perfectly, so that's why we didn't use all 600 nymphs, but it's pretty quick and efficient to do once you get the groundwork done. Then we also determined that Females are mating multiply because there are multiple fathers in the ootheca. The sperm doesn't get there magically. So this is a polyandrous system. And because females are mating multiply, that means males must be, well, they are contributing to ootheca. So you have 2 fathers, 3 fathers, or 4 fathers. Conservatively, we find out it's around 2, which means that with that sex ratio we have as you get further into the season, say you have 10 females, on average, if all those females are mating twice, you have 20 matings. If there's only 10 males, because the sex ratio is 1-to-1, that means there has to be remating opportunities for those males. And as males begin to die off because of predation or because of sexual cannibalism, so now you only have 5 males that have to make up for that 20 matings that the females produce. Their fitness is either going to increase or decrease if they get that 2nd, 3rd, or 4th mating, because they're not, if they get eaten after mating once, they're missing out on a 2nd ootheca, a 3rd ootheca, or a 4th ootheca. So depending on the actual rates at which they're mating, that will change how much their fitness is increasing or decreasing. Because if you avoid that sexual cannibalism and that early predation, and you last towards the end of the season when there's way less males around, you're going to have lots of females to mate with. And the females stay receptive to mating all the way up until they deposit their ootheca. So future work is just looking at all of this. I have some groundwork done, which I'll present at my thesis here in a couple weeks at Fredonia. But we were trying to look at inbreeding coefficients and the migration of rare alleles using the same techniques, those genotypes. As well as I was really interested in determining if there's any sequence in oogenesis. So I wanted to know, is sperm mixing throughout, so you'll see fathers evenly distributed throughout the whole ootheca, or if it's a layering system where the sperm isn't mixing at all. So if you're the first to mate, you're going to be at the top of the sperm storage receptacle. If you're second to mate, you'll be there, and then third to mate, because in insects, that matters. We have P1 versus P2, which is just, if you're the first in, you might be the first out when you're actually deposited. And there's also some skewing where if you're the first to mate, you'll have 70% of the offspring versus 30%. So I was interested in looking at that. And acknowledgments to all the people that made it possible for me to do this. And some citations. Questions?
Do females universally attempt cannibalism at every breeding?
No. So in my early workings there with Dr. Brown, I was interested in that too. It's a little harder to do behavioral work because you have to have them in the lab and everything. But I was interested, some females seem to be naturally more aggressive, so there might be some inheritance to aggression. But my advisor's done research on hunger levels. So if you have a really well-fed female, the rate of sexual cannibalism goes down. She's less likely to eat a male, as to where if you starve them, she's more likely to eat a male. And interestingly, he did a study. Basically, he would put a female on a stick. That way a male could only approach her in one direction, and they would have starved ones versus well-fed ones. They seemed to avoid the starved ones. The male was less likely to approach the female. And then they even changed the positioning of the female to face away from the male. That way she can't reach him or get him, and it's more likely for him to escape. They are way more receptive to that too. So orientation matters, and it seems as though they're willing to assess society or satiation in the female. How, we're not sure. It might be the levels of sex pheromones they're producing. It might just be a visual cue. That has to be explored more.
I got a quick follow-up.
Yeah.
Is it thought that the density of females might affect the male's, escape response? Like if there's only one female in an area and I'm a male, then maybe I sacrifice or try a little less to get away?
Well, it seems based on that study he was doing that males do have risk aversion behaviors. So basically a female will produce all these pheromones, and we'll see multiple males all approach the female. They won't all try to mount her, so some are less risk averse. So some will just go in no matter what. They don't care if she's gonna eat them or not. But there seems to be some that are like, nope, no thanks, I'll find another. So we don't know if that's like genetic. It might be an inheritable behavior from the parents. That's something we've thought about doing, but getting the live ones in is a little harder than the Ootheca, because I can just go around and rip those off shrubs and stuff.
Is there any evidence of sperm competition in this species? You may be touching on that a little bit.
In this species, it hasn't been very, well, in many praying mantis species, it hasn't been well assessed. Even I was, in my thesis writing, I was looking for reproductive structures in Tenodera sinensis, and there's like nothing. So there hasn't been any reportings of how many reproductive organs exist, their size and shape, because in ants and other insects, the shape matters. If you have a cylinder, it changes how the sperm mixes versus if you have just a sphere. There's been nothing on that. There's been nothing on sperm competition itself. So that's kind of why I started leaning into that towards the end of my research.
Hi, Sam. Good to see you.
Nice to see you too.
Sam, how common is multiple turning in insects? This is kind of something new to me. I didn't know that this was.
It's pretty widely. You can see it in a lot of different insects. In praying mantids specifically, it's more common than monogamy. So the rates, I don't have the rates off the top of my head, but it occurs more frequently than monogamy occurs.
We look at these insects very differently now.
Yeah. Thank you.
This concludes our first session of the day. We will reconvene at 2:05 in this room.
Yeah.
We've got like 30 seconds before our first presentation for this session. So I would like to invite Harjap Kathuria up. And this presentation is going to be on the prevalence and metabolic characterization of raw milk bacteria. And there was a little pointer, oh, it's right here. So this, you can advance your slides and point.
Is this the laser?
And you can point it. It should work.
I mean, I can use my mouse.
Oh, no, this one right here.
Just
There you go, the middle one.
Okay. Should I start?
Yeah.
Okay. Can everyone hear me? Oh. Hello? Hi everybody, my name is Rajab. Today I'll be exploring with you the prevalence of bacteria and the metabolic characterization in raw milk. Before we get started, I just want to throw a thought question out there. Has anybody ever wondered why we pasteurize cow milk but not human milk? They're made up of very similar things, and yet we all feel very uncomfortable drinking milk straight from the cow. Hopefully throughout this presentation you'll get the information you need to kind of make that decision on your own. So starting off, there are three main components of raw milk. First off, we've got macronutrients. This includes your proteins, your carbs, and your fats. We've got micronutrients, which include vitamins and minerals. And finally, we have a bioactive component of milk, which includes bacteria, enzymes, immune cells, antibodies, signaling molecules, things of that nature. These bioactive components, and specifically bacteria, can enter raw milk in various ways. First off, we have bacteria that are right there already endogenously within the milk. So before we even collect it, they're already there. Next, we've got bacteria that live in the environment of the cow. They can be on the surface of the udder, the bedding in which the cow lays on. All of this provides a source for bacteria to get into the milk. Third, during collection, this involves the cleanliness of the farmer, the equipment that the milk is being collected in. These are all reservoirs for bacteria as well. And finally, throughout processing, as we create dairy products, this equipment has its own set of bacteria that can also get into the milk. All right. These bacteria have many benefits for the consumer. First off, we've got competition. These bacteria can compete with pathogens to prevent or avoid colonization by these bad bacteria in our gut. These bacteria can help with digestion of milk. We have a lot of lactose intolerant people in this country. Bacteria can secrete an enzyme that helps break down lactose into its constituents, which are glucose and galactose. And finally, they can aid in protection or boosting the immune system. Regular exposure to these bacteria can kind of stimulate the immune system so that they defend better against other bacteria. Not all of these bacteria are good, however. You are twice as likely to have an infection or some sort of disease breakout by consuming raw milk. Between 2007 and 2022, there's roughly twice as many confirmed illnesses due to raw milk as opposed to pasteurized milk. However, you will notice that they have similar rates of hospitalizations, which suggests that although you are getting sick, the disease or the sickness might not be as intense. All right? Because there's bad bacteria, that's why we pasteurize. You don't have to worry about this, right? But the downside to pasteurization, this exposure to high heat, ends up removing or reducing the bioavailability of some of these nutrients. Specifically, we see losses in vitamins A, B, C, E, and some minerals such as iron, zinc, and calcium. Previous research up until this point has mainly been focused on determining which bacteria can be found in raw milk. So far, we've found over 250 species. I'm not going to bore you with all of those, but these bacteria have been grouped into 3 or 4 main categories. First off, we've got food technology and health promotion. These include lactobacillus, lactococcus, streptococcus, leuconostoc, and enterococcus species, and these are commonly referred to as lactic acid bacteria. Secondly, we've got spoilage bacteria, which includes Pseudomonas, Acinetobacter, Crassiobacterium, and Clostridium. And finally, we've got those that cause illness, which include Listeria, Staphylococcus, E. coli, Campylobacter, and Mycobacterium. Knowing which bacteria that are in milk is great, but knowing how they act and what they need is even more important, which brings us to the goals of our research. Our first goal was to figure out which bacteria can we actually culture. Them being present and us culturing them are 2 very different things. And secondly, we would like to know what roles these bacteria have as far as raw milk is concerned as for the consumer and in the development of milk products. So in order to test this, we started off by acquiring unpasteurized unspoiled refrigerated raw milk from a local store in Erie. We performed a serial dilution and plated that dilution to isolate for colonies. These colonies were then purified for 16S RNA, which is a conserved sequence among many bacteria, and were sent for sequencing to a separate laboratory. Our results came back with the majority of Pseudomonas species. This, along with Acinetobacter, are, as I mentioned previously, spoilage bacteria. We also found lactic acid bacteria in the form of Leuconostoc and Lactococcus. These are known for health promotion and food technology. And finally, we have potential pathogens, Citrobacter and Pterobacter. Potential pathogen, what this means is that if you do drink milk that has these bacteria, you won't actually get sick unless you have some sort of immunocompromised state or immunodeficient state. Sometimes even pregnant women, their immune system decreases in a way that they would end up having this contracting disease from these bacteria. Expanding on those the majority Pseudomonas species that we found, these Pseudomonas species can be split into 5 main groups. We have Pseudomonas aeruginosa, which are animal pathogens. We've got Pseudomonas putida, which are plant growth promoters. Fragi, which are dairy spoilage bacteria. Syringae, which is plant pathogens. And fluorescens, which are plant growth promoters. Our results came back with a majority of them in dairy spoilage and plant growth promoters, which is expected because these are raw milk bacteria. What we found curious was that this group on the right over here is called fluorescens. We were wondering, do these bacteria fluoresce actually? We shown them under UV light and found that AK5 and 8, the ones that are part of the fluorescence group, did indeed fluoresce under UV light. Moving on, we wanted to start categorizing the metabolic characteristics of these bacteria. We wanted to know what temperature range these bacteria can grow in, whether they're able to survive within the body or not, and which parts of the milk they needed to survive. Our method started out very similar to the previous state. Once we performed a serial dilution and isolated for colonies, we started performing various metabolic tests on them. I'm only going to show 2 or 3 of the main ones that we found, just for ease of understanding the research. We started off with checking temperature dependence. This is a lot to look at. I'm going to clean this up in a bit. Starting off, looking at our Pseudomonas species, you'll notice that over here, they're all cold tolerant. They tend to grow at 4 degrees Celsius and below. Whereas if you compare this to our remaining bacteria, they're not able to do that. And on the flip side, if we focus in on those bacteria, the lactic acid and potential pathogens, you'll notice that they grow at room temperature and 37 degrees Celsius. This is expected. 37 degrees Celsius is roughly human body temperature. The Pseudomonas species, although they're able to grow in the cold, they grow 3 times more slowly than they would at room temperature. This can be seen here by this growth curve. At 3 days, we have a absorbance of roughly 0.8. If you look at the bottom one, we're seeing it's taking 9 days to reach a similar number. Okay. The next test we ran was on McConkey agar. This is a agar that has biosalts. Biosalts select for gram-negative bacteria. The salts interrupt or interfere with the membranes of gram-positive bacteria, making it difficult for them to grow. Additionally, this agar has a pH indicator that turns pink when it's acidic, and it has lactose. So when we grow bacteria on this agar, if it can survive in the presence of biosalts, meaning it's a gram-negative bacteria, and it has the ability to ferment lactose, the production of lactic acid will reduce the pH, causing the color to change to pink. So on the bottom I have an example of this. Both of these bacteria are biosalt tolerant. However, only the left one is a lactose fermenter, and this can be seen by the color change. These are the results of our plating on McConkey agar. Again, this is a lot to look at, so I'm going to split this up. Looking at our Pseudomonas species, all of them were biotolerant. You can see this because they're all growing. However, because these are not pink, you can tell that they're not lactose fermenters. Our potential pathogens, all were also able to grow on McConkey agar, but they were also not lactose fermenters. And finally, the lactic acid bacteria were unable to grow. And because they were unable to grow, we can't infer whether they're lactose fermenters or not, because they weren't present there to do that activity. The next test we ran was on casein agar. This helps us decide whether bacteria can break down casein. Caseinase is an exoenzyme, meaning the bacteria release it into the environment. If these bacteria release it into the environment, you'll see a halo where it's digesting the casein, and that suggests a positive result. These were our results of this experiment. Again, I'm going to break this down. Looking at our Pseudomonas species, we found 3 of the 4. You can see a halo here, here, and here. 3 out of the 4 species were able to digest casein. For our potential pathogens, we don't see any halos, suggesting that they're unable to digest casein. And finally, our lactic acid bacteria were able to slightly digest casein. The last thing we tested was to see how these bacteria broke down various sugars. Our main focus, or the most important one really, is lactose, because that's the main sugar that's found in milk. If I focus on lactic acid bacteria here on the bottom, the second row, they're both able to digest lactose. The Pseudomonas species, however, except for one, were relatively unable to break down lactose. You will notice, though, that they were able to break down glucose and galactose, which suggests there might be some reliance on other bacteria to fully digest lactose, this milk sugar, because glucose and galactose are breakdown products of lactose. And just to wrap things up, so we wanted to figure out which bacteria were able to be cultured. We found mainly Pseudomonas, Acinetobacter, Citrobacter, Enterobacter, and lactic acid bacteria. We found that refrigeration slows the overall growth of bacteria, but tends to select for spoilage bacteria. In this experiment, we found Pseudomonas being promoted. Their ability to tolerate bile and ability to grow at 37 degrees Celsius suggests that these bacteria have an ability to colonize the gut. So if you were to intake them, whether they were probiotics or pathogens, they would be able to stay there. And finally, milk spoilage bacteria may require the presence of other bacteria to fully utilize milk sugars. This goes back to what I was discussing about the inability to break down lactose, but the ability to break down its breakdown products, including glucose and galactose. Some future directions for this research. In the very beginning, I mentioned that certain bacteria can be found within milk itself without even harvesting it. It'd be pretty interesting to figure out how this bacteria gets there. We have certain theories, but we're not completely sure how this happens. The relative risk of milk pathogens, if there are pathogens, milk also contains antibodies and other defense molecules so that the cow doesn't get infected, right? So if these molecules are in milk, what's the relative risk for us? If we drink it, are we going to get infected, or are these bad bacteria already inhibited by the molecules that are in milk? Commercial filtration. In laboratories, we've been able to separate out bacteria based off of what kind of bacteria there are. It'd be kind of cool to see a commercial application of this. Maybe we can select only for probiotic bacteria when we're, as opposed to pasteurizing milk. And then finally, alternative storage techniques. As you saw, when we refrigerate it, we select for spoilage bacteria. There are alternative storage techniques that might be able to preserve beneficial bacteria or even just the vitamins in general. For example, if you use an opaque container, you end up saving a lot of the light-sensitive vitamins, such as vitamins D and A. And with that, I open the floor to questions. Thank you.
Any questions?
Yes.
So for the raw milk, what is the shelf life? Like, how long can they sell it to people?
I don't know the exact date range. I assume there's some sort of standardized care, but there's no reference of oh, the bacteria have reached this certain point and now we can stop selling it. But usually, at least from what I've gathered from the research, the best way to consume raw milk is if you know the dairy farm directly, or if you live awfully close to one, that gives the bacteria less time to grow and proliferate. So you're getting most of the good stuff without as much of the bad stuff.
Any other questions? All right, thank you.
Thank you.
We still have a couple minutes before the next part of this session, so if you guys need to go to the bathroom or get a drink, you can do that.
We have Kara Hill on a lot of words I don't know how to pronounce, but she will say that for us.
Good afternoon, everybody. My name is Kara Hill. I'm from Mercyhurst University, and today I will be discussing fine dining in Erie, particularly some of the best restaurants. Just kidding. Of course, that was a picture of an edible coral fungus on an aesthetically pleasing plate. But it is very relevant because today I'll be sharing with you my research on the inhabitants of a clavarioid coral mushroom. So first, let's start off with what's known. We know that there are many bonds between fungi and other organisms. Oh, there we go. This includes relationships between fungi and bacteria. In this figure, you can see Candida interacting with Pseudomonas, and then at the bottom of it here, you can see Aspergillus interacting with Streptomyces. Streptomyces is a bacteria that looks like a fungus, and in this example, it is increasing the antimicrobial properties of the fungus Aspergillus. This is a fancy way of saying that it's protecting it from pathogens. So to narrow this down even further, what about mushrooms? Mushrooms and bacteria can also interact. Here we have brown blotch disease, and Pseudomonas thalassei is a bacteria that causes this. There's also another bacteria called Mycetocola, and it is preventing the virulence factors from Pseudomonas here from infecting it. So this is an example of a symbiotic relationship between fungi and bacteria as well as a pathogenic one. And how this whole research project started was when a couple of students went on a hike in Allegheny National Forest. They were initially looking for ghost plants, and they found ghost plants, but they were dead. They stumbled upon what they thought were ghost plants, but it was actually, upon closer observation, a coral fungus. So we brought it back to the lab to look into it more. While researching it, we identified it as a clavarioid coral mushroom. Here's some pictures from sources of these fungi. You can see that they're branched, they have fruiting bodies. They are also sapotrophic, which means that they grow on leaf litter and they prefer to grow on wood from fallen trees. They are also edible and they have medicinal properties. And interestingly enough, they are known to cause gastrointestinal issues when eaten raw. And this raises some questions. Is it something in the mushroom that is making people sick, or is it a bacteria that inhabits it? In the pictures over here, the bottom one is our actual coral mushroom that we brought back to the lab. And as you can see, the ones from literature look pretty similar to ours. And to answer our question, what bacteria colonize the interior of the coral mushroom, and is there a relationship between the two, the first thing we wanted to do was surface sterilize the mushroom. This ensures that we're extracting the bacteria that inhabit it, other than getting soil bacteria that might be on the exterior. Next, we did isolation with serial dilution plating, and then we sequenced our bacteria using the 16S ribosomal RNA sequence. And we ran biochemical tests, and then we tested the effects of our bacteria on plant growth. So this is our initial plate over here with all of our extracted bacteria that were inhabiting the mushroom. And upon isolation, we discovered 3 distinct species. They were unknown at this time, so we designated them as KH1, KH2, and KH3. Upon sequencing, we found out that we had 2 Pseudomonas species and 1 Presque. And in literature, we know that all 3 of these bacteria should present as gram-negative rods. So just to verify our sequencing, we performed gram staining. And you can see at the top, there's our colony morphology, and at the bottom, there are the gram stains for each. And sure enough, all 3 were presented as gram-negative rods. Because we had 2 Pseudomonas species, we wanted to verify that they were in fact different from one another. So we made a phylogenetic tree with our sequencing data, and we found one of our Pseudomonas species there, and then KH3, the other one right there, so they are different. They are both in the fluorescence group, and the Pseudomonas fluorescens group is known to glow under UV light. So to test this, we put our plate under ultraviolet light, and sure enough, they lit up. Next, we ran biochemical tests to discover a possible interaction between the fungus and the bacteria. So the important ones to point out is the citrate test, glucose, gelatinase, and caseinase. For our citrate test, we decided to run this because fungi utilize the citric acid cycle, so they will produce an abundance of citrate. It would make sense if the bacteria produce the enzyme to break down the citrate, because that could suggest a possible relationship between the two. And then all three of our species showed up as positive for this test, so all three of them were found to produce this enzyme. And we ran the glucose test, and only our Pseudomonas species produced the enzymes to digest glucose. Our Chrysciobacterium was negative for this. And then the gelatinase test we ran because gelatin is derived from collagen, which is found in abundance in coral mushrooms. And all three of our bacteria tested positive for producing the enzyme gelatinase. Then we ran the caseinase test using a milk agar plate. And all three of our bacteria showed up positive for this as well. You can see distinct halos around them. They produce the enzyme caseinase. And then lastly, we did the starch test, the amylase test using a starch plate. And our Chrysciobacterium was positive for this. Our Pseudomonas was positive, and then our other Pseudomonas was negative for this test, which just further helps us verify that both of our Pseudomonas species are in fact different. They even have different biochemical abilities. And I know what you're all thinking, there are crab legs, we're back to fine dining again. But we actually used these crab legs to make our own chitin plates. This is important to point out because chitin is abundant in mushrooms and our coral fungus. And it's a relatively difficult compound for bacteria to break down. So if our bacteria were to be able to produce the enzyme to break down chitin, that would be a strong suggestion that they are interacting with the mushroom. Here's me extracting, crushing up the crab shells to extract chitin from them. And we tested our plate to make sure that it worked by using a negative control and a positive control. We used Serratia, which is known to break down chitin. And you can see a distinct, pretty clear halo around this bacteria as opposed to the negative control. So this was very exciting to us. We tested this on our bacteria. You can see our controls in the corner, and then we have KH1, KH3, and KH2. And we saw a halo around KH3. Our Pseudomonas species was in fact chitinolytic, and we did multiple replicates of this. This was also very exciting to us because it suggests a possible relationship between the mushroom and the bacteria. Next, we tested our bacteria on plant growth to see if they would promote plant growth or maybe they were a plant pathogen. And we used our Pseudomonas to do this. And as you can see, it does in fact promote plant growth. We measured the root drive, weight per plant for quantitative data, and the data is significant. Our treatment group here is much higher than our control. And you can see the picture of one of our replicates. The control is on the left there, and our treatment plant is on the right. It seems as if the plant treated with our bacteria is able to bear the weight of the leaves on top of it. It stands up taller. So we concluded that it promotes plant growth. And the next thing we did was test our bacteria in cold temperatures. We were thinking the coral fungus would be outside, the bacteria would be outside. What happens when the temperature gets cold? Do they die off? Are they able to live? So we have a plate at 2 degrees, 0 degrees, and -2 degrees Celsius. And you can see our two Pseudomonas species there are able to grow in colder temperatures. And then of course there's our control at room temperature on TSA. This is my favorite slide. Yes, it's colorful, but it holds significant data. On the top row we have all of our bacteria on different plates showing their metabolic properties at room temperature. And then on the bottom, we have the same, except it's at 2 degrees Celsius. So we were wanting to know if our bacteria are able to be metabolically active at these colder temperatures. And they are. Our Pseudomonas species can ferment glucose at 2 degrees Celsius. It shows up positive for the citrate test, so it's able to produce citrates at 2 degrees Celsius, and it can even break down phosphate at 2 degrees Celsius. Something interesting that we found here, if you look at the casein test all the way on the right, casein's a milk protein, whereas all the other ones are sugars. It looks like our Pseudomonas species choose not to break down the casein at colder temperatures. Instead, it seems that they prefer to break down sugars at colder temperatures, and at room temperature, they will break down the casein, but at colder temperatures, they choose not to, so it can be inferred that maybe it's more difficult to produce these enzymes when under stress. For our conclusions, both Pseudomonas species utilize glucose and citrate, as well as casein as nutrients. And our Pseudomonas species, KH3, is chitinolytic. And both Pseudomonas species are able to thrive and utilize enzymatic properties at 2 degrees Celsius. And our Pseudomonas species seems to prefer to break down sugars at these temperatures rather than proteins. And then lastly, our Pseudomonas species, KH1, is able to promote plant growth. So I want to talk about the significance of this for both the agricultural industry and the relationship between the bacteria and the coral fungus. Could this bacteria be an opportunistic pathogen to the coral mushroom? From our cold tolerance tests and the fact that these bacteria are metabolically active at these cold temperatures could suggest that they are maybe an opportunistic pathogen. And an opportunistic pathogen is one that waits until the host is dying off or getting weaker, and then it attacks. So perhaps when it gets colder and the mushroom starts to die, then the bacteria take advantage of that to release virulence factors in this case and break down the coral fungus for nutrients. And then how would this affect the agricultural industry? Well, Pseudomonas is a broad genus of bacteria with species that do many things, but there are some that are human pathogens. There's Pseudomonas aeruginosa that can wreak havoc in cystic fibrosis patients. And then some Pseudomonas can upset your stomach and cause gastrointestinal issues. So if these bacteria are growing at colder temperatures, they're able to grow in your fridge. For future directions, we would like to do more with cold tolerance testing, maybe create a growth curve for cold tolerance testing so that we have quantitative data along with our qualitative data. And then testing for active lipase enzymes in cold temperatures. This would be significant because cold water washing for your laundry is becoming more popular due to the fact that it's environmentally friendly. And if we could test for the lipase enzymes that are used in detergents with cold water washing, this could be very beneficial. And then the last point here is quorum sensing for future directions. Quorum sensing is when a bacteria reaches a certain threshold level in its abundance and concentration, and then it starts producing their enzymes or virulence factors. And I bring this up because if we look back at that starch plate, KH2 or Cryptobacterium, you can see a clear halo that spreads out around the bacteria, whereas in KH3, it seems almost confined to areas in the bacteria that have highest concentrations of bacteria. And on the edges, you don't really see a halo. So this might suggest that the bacteria are waiting until they reach high concentrations to produce the enzyme amylase. This would be extremely interesting to test, and a lot of bacteria use quorum sensing when they infect a host to overwhelm the host's immune system. So this possibly could be going on in the coral fungus. An acknowledgement slide in there just to thank everybody who helped with the project. It doesn't seem to be in there right now But thank you all for listening. Oh, are there any questions?
I have a question.
Yes.
Is this coral fungi found in southern part of the United States in warmer areas? I mean, what do you think the bacteria how would it be different if it is found in warmer locations or?
That's a good question. I know that I know it can be found all across the United States. I don't know about temperatures near the equator, but that would be interesting to like research different samples from different places, see what kinds of bacteria are in mushrooms that grow at higher temperatures, because I wouldn't be surprised if they were different. It would make sense.
Or even if you think of the idea of in this region and climate change and things getting warmer for longer periods of time, that might change the interaction of the bacteria or what bacteria is there.
That would make sense.
Any other questions? Thank you.
We have a little bit of time, like 6 minutes before the next presentation, so. next presentation of the session, we have Isabella Presque on cactus roots harbor and endophytic growth promoting Pseudomonas species.
Hi, I'm Bella. I'm here with Mercy University, and today I am here to talk to you about plants. Now, plants are the most commonly utilized organisms in the world, arguably, and that's mostly because of their extremely unique and beneficial characteristics. First of all, they're one of the only known organisms that are able to use this energy from the sun to make their own food, which in turn provides food for us as humans and other organisms. It can also provide the materials for shelter and clothing and even medicine. So basically, in short, plants help us out a lot, but that begs the question, who helps the plants? Now, this question has been studied a lot in previous years, and one of the most common helpers of plants is actually bacteria. Now, bacteria have been found to be present in plants in various compartments throughout the plant. The three most important ones are the endosphere, which can be found throughout the plant. So the endosphere is basically just inside the plant. For example, there's the leaf endosphere, so it's just inside the leaf, the stem endosphere, and most notably the root endosphere. There's also the rhizosphere, which is the area immediately surrounding the roots, and the bulk soil. So bacteria can be found in all of these parts. But the most important ones are found within or around the roots, because the roots are one of the most important parts of the plant. So, who's exactly inside the roots? Now, there's four main phylum. There's proteobacteria. These are gram-negative bacteria. These are your Pseudomonas species. There's Bacteroidetes. These are gram-negative rods. This phylum of bacteria makes up about 50% of the bacteria that are present in your gut. There's firmicutes. These are gram-positive, and these are your, if there's any healthcare professionals here, you'll know it by the smell, C. diff, or type of firmicute. And then there's actinobacteria, which are also gram-positive, and this is your streptomyces. So why exactly are bacteria inside the roots? What are they doing there? How are they helping the plants? Well, they can help to absorb nutrients. For example, they can help to fix atmospheric nitrogen for the plant that the plant might be unable to do otherwise. They can also help promote plant growth, as you can see on the left there. And they can even help, in certain cases, fight against plant pathogens, as seen on the right here. So each plant has a specific set of bacteria that are present within the roots. It's unique to each type of plant, and it's dependent on a lot of factors, most notably soil composition and the host plant genetics. So, the root endosphere in many plants is not extremely well studied for specific plants, and specifically in the cactus plant, there's not a lot of research out there. So that's a major space for more knowledge to be gained. So first, to know what we need to learn about the cactus bacteria, we need to learn what we already do now. So in previous studies, the endospheric region of cacti roots have been found to be comprised primarily of fungi. What bacteria we have found in cactus roots have been known to help the cactus tolerate drought conditions and have even had a positive effect on various growth promoting abilities, including greater dry weight, root length, and height. Which brings us to the aims of this study. This study, we wanted to determine what the most abundant bacterial inhabitants of cacti were, and to determine what benefits these bacteria could be making for the cactus. So our methods include taking our little cactus friend. Ours we specifically randomly found on a beach in Delaware. Didn't know cactus grew in Delaware. We performed a serial dilution from 3 compartments, the bulk soil, the rhizosphere, and the root endosphere. For the bulk soil, just took the soil. For the rhizosphere, we took the soil that was on the outside of the roots, and then for the root endosphere, we crushed up the roots to get the bacteria that was inside. So we performed a serial dilution and plated our little friends onto plates, and from there, we did a number of things, including molecular characterization, morphological characterization, biochemical tests. We tested antagonism, which I'll get to later, that was a special surprise, and plant growth promotion. So to start with our results, we started with molecular characterization. So we performed 16S ribosomal RNA sequencing on all of our isolated bacteria. We had 18 total. I know this ends at 17, but that's because 18 was found inside, so it went back up to the top. The gene was amplified using PCR and sequenced using the 27F and 1779R primers. The sequences were assembled to form contigs and subjected to a nucleotide blast search to find the best matches. From there, we did colony morphology and gram staining. And here are all of our endophytes. You'll notice IF2 is missing. That's because on the previous slide from sequencing, we determined IF1 and IF2 to be the exact same bacteria. So we found that all of the endophytes were gram-negative rods, as you can see up here. From there, we quantified the distribution. We found that endophytes were mostly Pseudomonas species, as you can see by this giant circle. And gram-positive bacillus species dominated outside in the sand region, while the root outside was a better mix between the two, which implies that the cactus is selecting the Pseudomonas species while not selecting the bacillus species. So then we performed a phylogenetic analysis of our Pseudomonas species to see how they related to each other, and we found that they were closely related to the Pseudomonas fluorescens group. So we wanted to confirm this, of course. So we plated it on Pseudomonas F. agar and tested it under UV light, and as you can see, our little guy glue. So then we did a lot of biochemical tests, and I mean a lot. But we're going to focus on the important ones because that's way too much information for a 15-20-minute presentation. To start with, we did citrate, phosphate utilization, and gelatinase. So the citrate test, a blue color, indicates a positive result. That means that the bacteria can utilize citrate. Phosphate utilization was used on Pickup Sky's agar. Here on B, presence of a halo means that the bacteria can break down the phosphate present in the agar. And then gelatinase test is performed on nutrient gelatin. If there's a present of a liquid, that means it's a positive result. Then we also did salt tolerance tests on all of our bacteria. As you can see, it was just TSA plates supplemented with 5%, 7.5%, and 10% NaCl. Growth after 1 week was recorded for the presence of bacteria. Y is yes growth, N is no growth. So then, while we were originally plating our plates, we noticed that some of our little guys were eating into our other bacteria. So we took those ones that seemed to be fighting each other naturally and put them on their own plate, play bacteria fight club a little bit, to see if they would antagonize each other, and they did. So we performed an antagonism assay. So we put 20 microliters of a drop of an overnight culture of Pseudomonas IF3, is Pseudomonas alone, and then Pseudomonas IF1, and put E. coli between them, and then also put IF1 between them in the one test plate here. And as you can see, this is IF3 by itself. Here's IF3 with the E. coli, which was our control. And with IF1, you can see the growth is inhibited, but that's just visually, so we graphed it too. Quantification, we used image J to quantify the area of the bacteria, and a t-test was performed, and it indicated statistical differences. So from there, we did a couple of plant growth promotion tests. So we did 2 forms of these. The first one, we took the same little padlet, we cut it into 4,
and we dipped 2 of those into a control Pseudomonas IF1 suspension, a control suspension and a Pseudomonas IF1 suspension, and let them grow for about a week. And then after 2 to 3 weeks, we observed their sprout length, root length, all that jazz. We also did replica cacti, so we used different potted cacti and did the same thing with the control suspension and the IF1 suspension, and observed sprouts after 2 to 3 weeks. And then we quantified these with the number of new sprouts and the average weight per sprout. And the t-test confirms statistical difference, significant differences. So, our conclusions here. The uniqueness of Pseudomonas species inside roots implies that the cactus selects these bacteria to be present there. They have distinct metabolic characteristics, including citrate utilization, which suggests the plants may be sharing this citrate, allows for an adaptation of these bacteria to be able to use this as an energy source. The inorganic phosphate utilization suggests that the endophytes may assist the plant by breaking down this inorganic phosphate that the plants probably can't do on their own. And gelatinase activity may assist the plant in fighting off pathogenic fungi, as gelatinase is present in many of the fungi cell walls. Salt tolerance decreased in microbes as you move into the plant. It suggests that they are sheltered by the plant, so there's not as much of a need to be able to tolerate high salt conditions inside the plant as outside the plant. And endophytes have the ability to promote the growth and propagation of new cacti sprouts. So, future directions: the possibility of using Pseudomonas species IF1 as a cactus probiotic, and the possibility of drought tolerance benefits in IF1 and other endophytic bacteria. So, acknowledgements to these lovely people who helped me conduct my research, and to the Erie Community Foundation. Thank you for your time. Does anybody have any questions?
I was just curious, how did you determine which primers to use when you were doing that?
So we used the most commonly used ones, and honestly the ones that we had on hand, since funding, always gets a little bit crazy, so we just used what we did have already to make,
So you were basing it off of like a prior experiment that was done?
Yes, yes.
Any other questions? We have a little bit of time again.
So you talk about drought tolerance. If you compare the drought tolerance of the cacti to the drought tolerance of the bacteria that might be, with it there, is the cacti far more drought tolerant than the bacteria would be?
So we haven't exactly explored that yet, but previous studies have, which is why we're thinking about going in that direction possibly. Previous studies found that there's a specific, and I want to say it's Euphorbia trigonus, there's a specific bacteria in there that actually when it's present, it helps the cactus. The cactus is more likely to survive drought than without the presence of that bacteria. So we're looking in to see if any of these bacteria are doing a similar thing. So, yeah.
Interesting.
I'm just curious to ask, is it just one variety of cactus that could use this to prevent drought, or would it be different with a different variety of cactus?
Hopefully it'd be with a different variety of cactus. With our plant growth promotion tests, we used a couple different species of cactus. We started with the Opuntia cactus, which we found on the beach in Delaware. And then we started with a couple more from Lowe's to test plant growth promotion too, to confirm that it's not just one type of cactus or one specific cactus plant. So hopefully, we'd have to do more research into that, but hopefully we could confer that it would be for multiple species of cactus and maybe even multiple species of plants.
questions, thank you.
Got about 6 minutes before the last one.
All right, for our last presentation of the session, we have superoxide dismutase as a defense against oxidative stress in B. subtilis. And this is going to be presented by Jason Retker.
Thank you. I'd like to just say good afternoon, everybody. As you just mentioned, I'm going to be talking about superoxide dismutase as a defense against oxidative stress in Bacillus subtilis. I'm a junior at Mercyhurst University here, and I work in the lab with Dr. Mulholland and everybody else who presented today. So yeah, I'd like to start out by talking about honey. Now, honey is kind of revered as one of the only foods that doesn't spoil. So why is that? It's probably most likely, by and large, due to the fact that honey has an enormous amount of sugar in it. It's up to, I think, 70% of just sugar in honey. And really what this does is it causes a really hostile environment for microbes, inhibiting their ability to grow due to the osmotic stress from just having a sheer enormous amount of sugar in the environment. It pulls the water right out of the cells. And this prevents honey from spoiling. You know, microbes are really what's spoiling foods. If you think about cheese in the refrigerator, spoiled cheese is due to the microbe growth. I'm sure everybody's opened up the fridge and found cheese that has fungi growing on it. It's the worst thing ever. So yeah, what you would think is honey has no growth in it due to the fact that it has high sugar. In our lab, however, we were able to actually identify and find 12 species of super-tolerant, we'll call them bacteria, from honey. So there are some bacteria that can grow inside of honey. And we did this with honey that was grown here in Erie, PA. We crushed it up and diluted it and plated it out, and we ended up finding 12 species of bacteria. So like I mentioned a moment ago, these are all sucra-tolerant bacteria because they're able to tolerate the high sugar concentrations in their environments. And like I said, these are all environments that are enough to osmotically damage the cells. So if we think about it in the perspective of food spoilage, especially in the confectionery industry, lots of really sugary foods are obviously involved in the confectionery industry, and trillions and trillions of calories are actually lost every year due to food spoilage around the globe. That equates to about $162 billion lost globally just due to food spoilage, and this is with microbes that are able to actually grow in high sugar environments. So that makes us maybe more interested in explaining why my 12 bacteria are able to grow in honey and what kind of role they play in that. So with my bacteria, we actually were able to determine how much sugar they're able to grow in, how sugar tolerant they are. And if we map this onto something like E. coli, we can see that most of these strains are considerably more tolerant to glucose when on a plate compared to E. coli. And a lot of these strains ended up being Bacillus or Bacillus-related species. So I really wanted to look in and focus on Bacillus species. So there's a whole lot of literature about this subject in particular. So I was particularly motivated to explore this mechanism behind why these bacteria will survive. I was able to perform a SDS page gel with a few different Bacillus species to determine which proteins are being expressed at a 30% glucose treatment here. And as we can see with the negative treatment lanes, there is a lack of a 24 kilodalton band, and this is the commonality between each of these Bacillus species here. So this 24 kilodalton band, we sent it to Harvard for liquid chromatography, tandem mass spectrometry, to basically see what this is that the bacteria are emitting or producing to defend against high glucose. And we found that there's a lot of proteases at this spot, which makes sense. Bacteria will break down proteins to create osmolytes in the form of amino acids to combat osmotic stress. But what really made me scratch my head was superoxide dismutase, which is an enzyme that's usually used for combating oxidative, species in a cell, which I'll get to in a minute. But just to make sure he actually had superoxide dismutase here, performed a Western blot to confirm with SOD, superoxide dismutase, is commonly abbreviated SOD. So we did perform the Western blot, and we did confirm that yes, it is superoxide dismutase at 24 kilodaltons here. So like I said, it kind of just made me scratch my head as superoxide dismutase is involved, specifically manganese superoxide dismutase is involved in the reduction of reactive oxygen species, as you can see here, into hydrogen peroxide, which is a less toxic chemical for the cells. Yeah, so we did this Western blot with the rest of them, or a lot more of these bacillus and bacillus-related species, and we can just see there's a lot of signals here for superoxide dismutase. So conclusively, superoxide dismutase, SOD, is very important for high sugar environments for these cells. So, like I said, it made me kind of scratch my head as to why. Like mechanistically, why are reactive oxygen species and the defense it gives them in the form of SOD so important and so damaging to cells, and how does that relate to glucose? Well, it turns out that once I dug into the literature more, high glucose really just generates reactive oxygen species as a result of metabolism. A lot of these reactive oxygen species come in different forms here, as we can see, and this is, like I said, reduced or created by the process of metabolism. In glycolysis, which is the breakdown of glucose and for energy, in the pentose phosphate pathway, as well as the citric acid cycle, we can see reactive oxygen species play a role in inhibiting and also are produced by glycolysis and these processes in general. So it makes sense as to why glucose involves reactive oxygen species, as the metabolism of glucose itself actually produces reactive oxygen species. So in humans, you go to the supermarket, you find foods that say antioxidants, foods that are high in vitamin C, for example. Vitamin C is a very nice antioxidant. It helps reduce oxidative stress on cells and in humans. So foods like pecans, strawberries, or black beans are all antioxidant foods. And what that basically just means is that they prevent against reactive oxygen species. Reactive oxygen species are a major problem for bacteria, as we saw earlier, but also for humans. Just to name a few of the diseases associated with oxidative stress, we have cancer, diabetes, cataracts, neurodegenerative diseases, cardiovascular diseases, pretty much you name it, ROS might be involved with it. So I think a clear distinction could be made with the rest in peace here on this too, so maybe it should actually say ROS for reactive oxygen species. Yeah, and in conclusion, reactive oxygen species are just very damaging to cells and humans. So we wanted to actually make sure that superoxide dismutase is important for growth of bacteria, specifically bacillus species on high glucose media. So we were able to acquire a genetic defect, a bacteria strain that has a genetic defect compared to its wild type in the superoxide dismutase gene. And what we found is when compared to the control treatment, a 40% glucose treatment will result in a inhibition of growth in the mutant as compared to the wild type bacillus. So at this point I was wondering what other stresses might cause reactive oxygen species apart from sugar, because sugar is all great and all, but really in the body and just in general, there's many chemicals that come into contact and involve biochemical pathways with bacteria and humans alike. So at this point, we were wondering about stresses, and I really just wanted to focus my project just on SOD, just on superoxide dismutase and how it reacts in cells and in humans. So we did just that. We found different stresses that had the same sort of response, creating reactive oxidative species. We can see the control here. The control treatment between the wild type and the mutant resulted in no clear difference, no difference at all really between the growth when there's no stress. As you can see, 30% glucose created a clear decrease in the growth of the mutant. 8 degrees, which is about fridge temperature, saw a almost complete loss of growth with the mutant as compared to the wild type. And the same thing sort of for 55 degrees. You can see inhibition of growth here. And with 7.5% NaCl. I included this on here even though there's not a clear difference between these two treatments because of the next experiment that I performed, which was the Kumasi blue stain of an SDS page for the differential protein expression between the control and the different stress treatments, as you can see here at the top, between the wild type and the mutant. So we can see here in the control vein, remember we're looking at 24 kilodaltons-ish. We're looking for superoxide dismutase around this spot here. And as we can see, compared to the control, all of these stress treatments, wild types, involve the enzyme at that spot, at 24 kilodaltons. Conversely, the mutants do not possess the ability to produce SOD, and thus they don't have a spot there. They don't have a protein being produced at 24 kilodaltons. Yeah, and that's 24 kilodaltons. That's the manganese superoxide dismutase molecular weight. So just to confirm that these spots are actually SOD, because there could be other proteins like we saw before, like proteases and other things like that. We performed a Western blot with the certain treatments. And as we can see, remember from earlier, my salt plate didn't necessarily show inhibition of growth, but in fact, SOD is used in the wild type when it isn't in fact involved in and is exposed to high salt concentrations per se. And really what that says is we just need to up the stress if we want to visually see inhibition of growth on a plate. So we're going to have to maybe go to 10% salt or 12.5% salt on a plate to really see this. But this doesn't subtract from this idea that there is some 24 kilodalton bands on these different stresses just on the wild type, which is confirmed that it is superoxide dismutase. You might be wondering why there's not bands here and in the control. This is a part of the research we were trying to perfect. Western blots are notoriously difficult to nail down, get correct, and it's really just, there's so many things that can go wrong, really. So moving forward, I really want to focus on perfecting the Western blotting process. But this gives us a general idea as to locations of superoxide dismutase in the Western blot at 24 kilodaltons. So in conclusion with my data so far, we found sugar-inducible proteins. Remember the proteases from earlier that explain the osmolytic sequestration of osmolytes, really, just to prevent against the osmotic stress. And SOD, which mentioned is for reactive oxygen species, to defend against reactive oxygen species. This, like I said, this is all done by SDS page and liquid chromatography, tandem mass spectrometry. And we also were able to find SOD being acquired in various stresses. Remember, glucose was one. We had heat treatments, salt treatments, and cold treatments that all found SOD as an enzyme that is required, or at least related. And this is confirmed by Western blotting and the fact that the mutant knockouts were defective. So in the future, I really want to look into more stresses that have oxidative implications, like ethanol or hydrogen peroxide. As we saw this week, I actually streaked out a mutant onto an ethanol plate, and we can see a clear inhibition of growth in the mutant for the SOD mutant, and that's on the 10% ethanol. Hydrogen peroxide is something I need to perfect. I need to get the concentrations right. But in the future, I really want to continue looking at ethanol, hydrogen peroxide, and potentially do a Western blot and look as to see if SOD is there as a signal, because they're releasing oxidative stress. Also, I'm looking into paraquat, which is not necessarily industrial, it's more of a commercial herbicide. It's something you could buy at Lowe's and go spray. It's a weed killer. And I think that this is particularly important due to the fact that, one, the paraquat itself is reactive. It creates reactive oxygen species due to reduction oxidation chemistry. And the fact that right now we're in a watershed that affords most of the runoff ends up in Lake Erie. And globally, it's known that paraquat is damaging to freshwater fish. There's not necessarily any studies done as to paraquat in Lake Erie, and what effect it has on the freshwater fish here, but it is known that freshwater fish are in fact damaged by paraquat. So what kind of role does SOV play into this? Maybe if we can find some sort of, if we can understand the methodology behind bacteria that can survive in high paraquat concentrations, we can predict the outcomes of paraquat ending up in lake Erie as an environmental pollutant. And also in the future, I'd like to just think about the medical implications of all this. A lot of research recently has to do with the gastroenterological connection between bacteria in the gut and the actual health of a human. So if we can understand the role of superoxide dismutase in the gut, then maybe we can understand and better explain ailments such as cancer, inflammation, like I mentioned before, and the relationship between bacteria in our gut and in humans. And moving forward with that, I think that there's a considerable connection that might be able to be made, considering that there are the same processes involved in bacteria, like the breakdown of glucose in glycolysis, are also involved in humans. Potentially, the research being done on superoxide dismutase will have implication as to enzymes that can be produced to directly fight in a human cancer and all the other ailments I mentioned earlier. Here are my references, and I'd like to thank the Pennsylvania Academy of Science for funding for this research, and some of my fellow undergrads in the crowd here, as well as Dr. Mohan, my research advisor and mentor. Thank you. Are there any questions?
If there are no questions, then that concludes our second session. Thank you.
Thanks.
Yeah. the aquatic biologist with the Regional Science Consortium, and I'm going to announce our speakers for this session. So our first presentation, oh, they haven't brought me the things yet, actually. Yeah, sorry. Thank you. I won't announce that they don't have to stand up here. I'll stand up here. So our first speaker, it's going to be a speed talk. It's going to be the role of PCBs in the alteration of inflammatory markers and genetic changes in breast cancer by Trinity Priestash. There is a clicker right here if you want it.
Okay, thank you. Okay, so to start with a little bit of background, polychlorinated biphenyls, also known as PCBs, are contaminants that are found in Lake Erie. As ships go through, they dredge up these PCBs, which are consumed by microorganisms, and then through the food web, they can make their way into game fish, which is consumed by humans. PCBs are a known carcinogen and can be linked to cancer development, including breast cancer. And Erie County women are in the top 26% of breast cancer incidents in the United States. So our question was, could the consumption of game fish and therefore exposure to PCBs increase the rate of breast cancer in Erie County women? So my project was an expansion of a seed grant headed by Dr. Keyes, Dr. Newtile, Dr. Simpson, and Dr. Russell, where they were looking to quantify these PCB concentrations in game fish from Lake Erie, then model this assimilation in human tissues, and determine if the PCB exposure could promote tumorigenic changes in cell lines. My project specifically was focusing on the extracellular vesicle component. EVs are 40 to 500 nanometer vesicles that contain proteins, lipids, and nucleic acids. EVs are released from its host cell into the extracellular space, and they contain mRNA. So if this RNA is altered in any way, it can be picked up by a recipient cell and possibly change its phenotype, which is EV's role in cell-to-cell communication. This cell-cell communication can play a major role in cancer development. As these EVs are released from cancerous cells, they could contain apparently expressed proteins or nucleic acids, and if they have abnormal RNA expression and therefore incorrect protein expression, they could change another cell into a cancerous phenotype. So if a host cell has a cancerous phenotype and alters its RNA, that EV can be released into the extracellular space, picked up by a recipient cell, and then promote that cell to then change into this cancerous phenotype, which would be the spread of cancer and could contribute to cancer metastasis. So we were looking to see if PCB had involvement in this process. Our long-term goals of the project were first to look at the cellular expression and determine if there are gene changes in the host cell after PCB exposure. If we did see genetic changes, we then wanted to look at the extracellular vesicles and see if they were also promoting these abnormal gene changes. If we find these things to be true, was there a causative relationship between PCB exposure and the onset or metastasis of breast cancer? And then if we see abnormal gene changes in extracellular vesicles, are they passing these traits onto another cell, contributing to metastasis, which would be the cell-cell communication aspect? And if we see this cell-cell communication happening, we wanted to look at the role and mechanism behind it and see how these EVs are actually passing these traits along. So our methods behind this, we cultured NIH3T3 cells. NIH3T3 cells are a fibroblast cell line that we use to induce or transform a cell into a cancerous phenotype. We conditioned these cells under 3 different conditions. First was our vehicle control, or a DMSO sample. It was used to then quantify that we didn't have any results due to the DMSO because PCBs and Taxol were both reconstituted in this DMSO as well. Our second condition was the PCBs, which was quantified with a concentration determined from the seed grant. And then third was our Taxol, which served as our positive control, as Taxol is known to promote a cancerous phenotype. From this, we then isolated our extracellular vesicles from conditioned cell culture media via size exclusion chromatography. We first used differential centrifugation to get the larger debris to the bottom, and then we took that supernatant and using ultrafiltration, we took it from 15 mils to 500 microliters, and then microliters, we put over a size exclusion chromatography column, which separates particles based on size. Our first samples that come out are our void samples, which have nothing in them. And then next is our extracellular vesicle fraction, followed by our extracellular protein fraction. Our 4 EVs and our 4 protein fractions are then combined into one. So we had 1 extracellular vesicle and 1 extracellular protein per condition. We then characterized our extracellular vesicles using Western blotting. We were looking to examine the expression of CD63, CD9, CD81, and flotilin-2, as these are all known proteins to be expressed in extracellular vesicles. We did this to properly show that we isolate our extracellular vesicles and that they were present in our samples. From this, we then isolated our RNA to make cDNA. With our cDNA, we used RT-qPCR to assess gene changes. Specifically, we were looking at TGF-beta, VEGF, CXCL12, P10, P53, and CCL5, as these are all known gene changes that occur in breast cancer. Our Western blot results showed that we did get extracellular vesicles in our samples. The picture on the left is our CD9 results. We have ladder followed by PCB extracellular vesicles, and then the PCB protein samples, and then DMSO and Taxol. As you can see, we do have bands showing up for CD9 in all the extracellular vesicle samples and none in the protein fraction, which showed that we did have extracellular vesicles present in those samples. We followed that up with flotilin-2, which also confirmed the same results, that we have flotilin-2 in the extracellular vesicle sample and not in the protein fraction. To further confirm the presence of extracellular vesicles, we did send out samples for electron microscopy. This is a 1:10 dilution of our DMSO extracellular vesicles, and all of those right there are extracellular vesicles present, so we can see that we did have an abundance of these in these samples. Our PCR results, we didn't really see any significant changes across the culture conditions. This graph here shows our P10 PCR results. As you can see, weeks 1 through 3, there's not really a significant change in gene expression for P10. In week 4, we do see a 10-fold change in gene expression for the overexpression of it in the PCB sample. We cannot really conclude anything based on these results because there was trouble with RNA quantification. Our nanodrop was not accurately quantifying our RNA, so there could have been an incorrect amount of RNA put into our CNA, which may be why we weren't seeing significant PCR results. So we would like to redo these samples with the proper RNA quantification to see if we do see genetic changes in the fibroblast cell line. But another reason that we may not have been seeing significant changes is that we were targeting breast cancer specific genes. Since we were targeting these genes, we may only see them in an actual breast cancer cell line. So we would like to further do this research with an actual breast cancer cell line. So moving forward, this is going to be an ongoing project throughout the academic year. In addition to redoing the PCR on the NIH 3T3 cells, we would like to look at gene changes in the MCF7 cell line, as that is a breast cancer specific cell line. And we'd like to target the same genes and see if we can see any changes across those with different exposures. We would also like to assess the NF-kappa B inflammatory pathway with PCB exposure. We know that chronic inflammation can also lead to breast cancer. So if these PCBs are causing the NF-kappa B pathway to be activated and causing inflammation, it could eventually lead to breast cancer, which would also show that PCBs do have an involvement in the onset of breast cancer. I would like to thank Penn State Barron and the Shires Honors College for this funding opportunity, Dr. Newtile, Dr. Simpson, and Mr. Cody Wood for concentrating the PCBs and figuring out the correct concentrations, Neshva Ossoy for culturing the NIH 3T3 cell line, and Dr. Ashley Russell for overseeing this entire project. Thank you. Is there any questions?
Do we have any questions?
So when you were showing the different weeks, on week 4, the PCB was much higher. Did it take until 4 weeks for that to be detected, or what would you expect if you went to week 5, 6, 7?
We would like to see the same thing. It's hard to say because P10 is a tumor suppressor, so if it was causing cancer, we'd expect it to be underregulated, not overregulated, which is kind of why we were thinking that the cDNA may have been off due to the RNA quantification. So that's kind of why we want to redo our results to ensure that that is correct.
Okay. Any other questions? All right, thank you very much.
Thank you.
All right, so up next we have Rhiannon Toy and Chloe Christ giving a face to the unknown, biometrics applied to the identification of human remains.
Hello, my name is Rhiannon,
I'm Chloe.
and we're both students in the Mercyhurst Applied Forensic Sciences program.
Our project is on giving a face to the unknown, biometrics applied to the identification of human remains. This project was led by Dr. Sarah Zapico and Dr. Joe Adzarius-Garriga. Dr. Adzarius is our professor at Mercyhurst, and Dr. Zapico is at the New Jersey Institute of Technology.
So a little bit of background before we jump into the actual project. The part of the project that we worked on was the forensic anthropology aspect. And if you didn't already know, forensic anthropology aims to utilize osteology to help identify deceased individuals. And if you don't know what osteology is, that's the bones. And approaches to ID vary, all with their pros and cons, which is why we're looking at implementing a multidisciplinary approach. So our proposal is, how does a multidisciplinary approach change the likelihood of identifying deceased individuals? And with this, we wanted to see if by utilizing a multidisciplinary approach, we can balance out the pros and cons associated with each individual method, increasing the overall accuracy and likelihood for positive identifications.
So first what we want to do, since we don't have the ante-mortem data, we start with the post-mortem data. And there are 4 things that we looked at for this project. First is a computerized facial approximation, which is a forensic artist does this, and they look at cranial landmarks and tissue depth markers to try and reconfigure what the face looked like. Secondly is the single base polymorphism analysis, which is using DNA to reconfigure the exterior visual characteristics and the biogeographical ancestry. And then epigenetics is also using the DNA and is trying to look at age at death. And lastly, what we did was the biological profile. what is a biological profile? The biological profile is composed of 4 things: sex, age, population affinity, or in more common nomenclature, ancestry, and the stature. As you can see, we are missing one. For this research, we decided not to do stature because it didn't work with our proposal, which was to reconfigure the facial characteristics.
So these are the 4 individuals that we primarily worked with for this project, and they're part of the Mercyhurst skeletal collection that we have, which is used for teaching purposes or research projects such as this one. And moving forward, we're going to talk about bioprofile. And now the bioprofile for these individuals are already known and recorded in the face sheets that we have in our department. But so we could get an unbiased feel for bioprofile, we did it from a blind perspective. The first method we used was a non-metric method, which means we didn't use any calipers or anything. It was just based on visually seeing things. And it was the Clels and Cool method using MorphoPass. And MorphoPass uses morphological traits of the skull and the pelvis to estimate sex of an individual. So the 4 traits that we worked with for the skull were the supraorbital margin, which you can find just by pinching just lateral of this little foramen right here. And it tends to be thinner and sharper in females and thicker in males. And then if we move to the side, we have the mastoid process right here, and it tends to be more robust in males. And then we have the glabella, this smooth portion in between the eyebrow ridges, and that also tends to be more pronounced in males. And then if we move to the back, we have the external occipital protuberance, aka the nuchal crest, and it also tends to be more pronounced and robust in males compared to females. But this isn't always the case, as Chloe's going to get into with the pelvic traits.
So when it comes to the pelvic traits, they tend to be more robust in females because to account for the realities of females having to give birth. So first we want to look at the ventral arc, which is going to be on this plane here, and it's going to be more of an angle and more robust in females to account for that widening. And then just inferior to that is going to be the subpubic contour, and then this is going to be thinner looking at it from this angle and concave in females to account for the widening up here. And then in males it's mostly absent or, and then if we turn it this way here, just under the pubic symphysis, and now when you look at this in your body, pubic symphysis is going to be anterior, and then this is where the sacrum rests. And so if we look under the pubic symphysis, there is a slight pinching, which is the medial aspect of the ischiopubic ramus. And so if it's sharper and more pronounced, then it's likely female, and dull or absent in males. The next method we looked at was using the TA3 software with Boldson, and this is for estimating age. And so first, just to get a baseline, we use the cranial sutures here. We'll see the coronal, and then down the middle here, the sagittal. And these traits are vestigial of growth and development. As we know, when we're infants, our cranial bones aren't completely fused. And we see this throughout our lifetime. And so when we know about what these fusions look like throughout our life, we can estimate at what age the individual was at.
However, those traits are very variable, so we don't want to rely too heavily on those. Just use it to get a baseline, like Chloe said.
Yes, which is why we then move back on to the Ascot set. And what we want to look at here is the pubic symphysis, as I mentioned before, and then the auricular surface where I said the sacrum sits. And what we're looking at is the topography. And so we score these qualitatively using non-metric methodology. And what we want to look at for the pubic symphysis is there any lipping? Is it flat? Same kind of for the auricular surface. Is there any billows? And then depending on what we see, we score them and plug that into the TA3 software, and it'll give us a statistical analysis of the likelihood of the age of the individual. And thirdly, the population affinity. For this, it was completely metric methodology. We used 24 cranial measurements between 2 or more perennial landmarks to estimate the population affinity. And for this, we use Fordisk. In Fordisk, there are several populations programmed into it, and so when we enter our data, we choose the parameters for kind of what we have an inkling of what the ancestry is going to be, just because there is such a wide range of population affinities in Fordisk.
In terms of results, 2 of our results are still pending, but the 2 individuals that we do have results for came back as Caucasian males, which honestly makes a lot of sense considering the majority of the skeletal collection that we have is made up of Caucasian males. And we don't pick them, they just come in, that's just the way it is. And then for MD20003, it fell in the age range of 40 to 85. And then MD21003 fell in the age range of 20 to 45. And now you might be wondering, why do you have such a broad range for age? And that's to account for the variability of the remains, because skeletal traits can be very deceiving and misleading by things such as nutrition, illness, like stuff like that, anything that can really affect the overall bone structure. And that's why we rely on different methods too. So we're not relying heavily on just one method. In terms of next steps, we took bone samples out of the femurs of all 4 individuals and we sent them to the New Jersey Institute of Technology so that they can carry out the next phase of this project, which is going to be the forensic DNA phenotyping.
Thank you. Any questions?
We have plenty of time for questions, if anyone has any questions.
So when you're doing some of those measurements, like you said, in the skull and looking at how thick or thin things were, during that part of the presentation, and you were saying it kind of defines if it's a male or female, can you see that in younger aged, or is it really hard to tell and you can only tell like once they've reached maturity?
In juveniles, yeah, it's very hard. We usually don't.
So you can't.
Juvenile males especially can have more feminine characteristics just because things are still developing.
So we usually don't in juveniles.
And then when you're scoring it, do you have more than one person score it to come up to the result or the answer? Do you have like multiple people scoring?
We checked each other's work, and then
We just had to make sure that we were kind of within the same range, because obviously we don't want somebody to look at a trait and be like, that's a 1, and then the other person be like, that's a 5, because that's an issue. That doesn't line up.
Okay, thank you.
All right, we're going to have a brief pause, and then we'll start our next presentation at 4:10.
Alrighty, we're going to get started with our final talk for this session this evening. And this will be the contribution of scene recovery data in skeletal trauma interpretations by Joe and Sirius Garriga. Oh, good.
Well, thank you.
I was told that it works.
Okay. So thank you, and thank you for saying my last name so good. I didn't expect that. Normally it doesn't happen. So I'm Joe. I'm an assistant professor at Mercyhurst University at the Department of Applied Forensic Sciences. And the ones that are not very familiar, what we do at Mercyhurst is we do a lot of casework, like lots of them. In the last 2 weeks, we were dealing with 9 different cases, and we were like, at the same time, we are giving our classes and we are doing our research, and we do all the casework and we do all the research projects at the same time that we are training our the next generation of top forensic sciences. You have seen 2 of them right there. So when we do casework or when we are called for a case, it can be something as like Dr. Dirkmatt receiving a text message from the authorities saying, hey, is that human bone or it's not human? If it's not human, call it a day, and we can continue with our classes, with our research, with our normal schedule, if we have one. But if it's human, then we have some more work to do. So our cases can be like from the search and recovery. So we will collect the human remains and the evidence for the authorities. The human remains, we will take them with us to further study or further analysis, and then all the evidence or the physical evidence that it's not like cell phone or backpack or shoes or whatever, then we will give it to the authorities. So regarding the human remains, when we transported them to we have like a morgue at Northeast, we do all the processing of the body, and then we proceed to the osteological analysis, which will entail basically the biological profile that you have seen with Rhiannon and Chloe. And also an important part of forensic anthropology analysis, or the forensic anthropology capacities nowadays, is the skeletal trauma analysis. So this is the 2 main things that we will do. We will do the dental analysis. The dental analysis will contribute for the biological profile in terms of age. We don't estimate sex with dental information. We don't estimate stature with dental information, but they are very good for age estimation, especially in juveniles. And of course, dental data will be very, very useful for personal identification. So sometimes we are at the scene, and the first thing there I ask for, like, hey, please give us dental records, because sometimes in like less than 24 hours, we have identified the individual, they can reach out the family, and then so on. If there are some trauma signs in that skeletal remains, the identification is made so quickly, then the trauma analysis, it takes a long time, okay? And then we do the taphonomical analysis, which is like, why are the remains dispersed like in that way? What are the different factors that have affected the remains once the individual was dead? So once we are like talking about the post-mortem timing. So as I said, like the trauma analysis is really important. And since we had the addition of dental identification, the identification of the remains have expedited a lot. And the taphonomical analysis is something that it's like all the taphonomical analysis, or how we manage the scene and how we interpret the data of the scene. I think it's something unique that Mercyhurst has, and that's because Dr. Dirkmatt is like the reference on this. So we are very lucky to be here. So if we do not take good records from the search and the recovery, especially from the recovery, it's impossible that we are going to do a good osteological analysis and especially good taphonomical analysis. It results impossible. And I bring you here 2 cases to show how we approach the cases, how we approach the from the scene recovery until the issue of the results in our reports. So we have learned by Dr. Dirkman how to apply the forensic archaeological methods in the proper way. That way you are not going to lose information from the scene, and that's so important because once we start to process the scene, once we start to alter the scene, this is gone forever. So if you don't get good records, you have lost forever this information. But the most important thing that we have at Mercigus, and like that's because Dr. Dickman's approach of the scene, is that we integrate. It was buried remains in Pennsylvania, but we did not recover the remains. So the remains were recovered by another agency, and they were brought to our laboratory at Northeast. So this is how they look when we opened the body box. So there's a lot of adipocera that it's very commonly formed when we have like a wet environment. And you can see the cranium here that was disarticulated from the rest of the body. So we had like one of these bags contains the cranium, and all of these it's the rest of the body. So after we processed the body, that's how it looked like, and we can see the cranium here, and we can see that there are like some defects over here. So we have some perimortem trauma in the cranium, and if you look at the radius, at the right radius, we have like also it's broken. Okay, it has a fracture. So we had perimortem trauma in the cranium and in the radius. So if you look at the cranium, what we had, there were some gunshot defects. We can see that one, that it's a very beautiful one. It's an entrance, a clear entrance defect. We have the beveling inwards. Opposed to that one, that we have the beveling outwards, and that means that will be an exit defect. We have this one here that we are missing some parts of it, so we didn't recover like all these small pieces. And also we had this defect here that I remember that some people had like the discussion if that was an entrance defect or it was like a fracture that was kind of awkward along the suture. So something that I do every time that I see defects is that I'm a forensic odontologist and a forensic anthropologist, and I take dental radiographs to the defects. So you can see all the bullet wipe here, and it's like there's no doubt that this is an entrance or an exit defect. So those are like, if you see, we have like some fractures that come from the defect. So that's what we call radiating fractures. We can have radiating fractures or concentric fractures associated to the defects by gunshot trauma. But if you look at very carefully, like this fracture here, this fracture doesn't look like all those ones. Look at the edges. Look at the coloration of the bone. So this is generally what we will classify as post-mortem damage. Okay, and we're like, okay, so why is that here? So one of the agents says, by the way, doctors, I was doing the recovery and I put a matic on the cranium. So that's how I found the cranium, which is very unfortunate, right? So we had to define very well what was the post-mortem damage, that you can see that here in blue, and take it out of the equation to focus on the pre-mortem trauma. So this is like, so this is like example of how not to do recovery, but it's one of my favorite cases because you can see like very, very well the pre-mortem trauma and the post-mortem trauma actually. So for teaching purposes, that was like priceless. We could see, depending on how the different fractures intersect with each other, we can have like the sequence of the different defects. So we can have like the first event was here, and so all the fractures are not interrupted by anybody or any other fracture. And then we have this second fracture that you can see like the radiating fractures, they end up in that one, so that means that this one was first, and this one here, it ends up here, so that one was done before this one. So we have the first event, the second event, and the third event. If you see, if you look at the cranium, there's like that one here that we do not know it was first, it was the second, because we don't have any fractures intersecting anyone. So we have to take that out of the equation for the sequence. But that one was an entrance defect that was very interesting because we really could see that it was an entrance, and inside the cranium we had the clinotes process. So it's like something that is in the base of the cranium, and it will be like, if that's the cranium, that will be like right here, and we have the defect here. So those can be like damaged because of the same bullet and in the trajectory. And then we thought like, okay, what's the exit of these projectile? So do we have it like from the foramen magnum, that it's like a very big hole that we have, a very big foramen that we have in the base of the skull, or is stayed like there inside the cranium, and we don't have any exit defect. We didn't know that because we have not recovered the cervical vertebrae. So if the foramen magnum was like, if the bullet was gone to the foramen magnum and did not impact the data here, we cannot know if that was the case of the trajectory or not, because we had not recovered the cervical vertebrae that were key for that interpretation. And also here we had the rate radius, and we said like, it's broken, it has a fracture, it looks like a gunshot, but we really don't know. What did I do? Take the dental x-rays, and we had like one bullet wipe. Because we were missing so much of this radius, we could not see like very well the direction, the trajectory, if it was an entrance, or it was a part of an entrance, part of an exit, and so on. So we decided to go back to the site and do a second excavation, this time done by Mercyhurst University scene recovery team. And that's what we found there. So they left quite a lot of behind. Especially we had this vertebrae that was like super important for us, and the distal radius. So we could have like a more complete skeleton. So first thing that we had is these verts were very nice. They had no damage. They had no perimortem trauma by gunshot. So we knew that that bullet was stayed inside the cranium and was one of the bullets that we recovered in the recovery. And then all these parts that we recovered from the radius, look at what nice. So we just had that at the beginning, and now we had all these. We could see like it's an entrance defect, and we could see like the direction, the trajectory, etc. So that was much more information than the one that we had. So in this case, we can see like all the details about the perimortem trauma by the gunshots were clarified, and that's the importance of the recovery. The second case that I brought, it was a case that was in April from 2021, and I remember that we were doing one case of scattered remains, and then Dr. Rigmert had a phone call, and he said, okay, hold on, we have to we have to finish this scene, and then we will go like back to, in our way to Erie, we will go and stop and do that scene. So we had this scene, that's how it looked like. So we could see a fire pit, and those legs were very, really intact. So all from here on, it was absolutely calcination. And the first thing that we have, it's okay, Dr. Digmet says how we have to approach, how we are going to record all the information, and then we recover the remains, applying proper archaeological methods. And we took this hand draw map, and that's very important. Nowadays we scan the scene, we take points, we map digitally. It's so cool, but we always take hand draw maps just as a backup, just in case. And this is how it looked like. And this what are the unburned bones, that they were everything that was outside of the fire pit. So from this case on, I'm absolutely certain that these fire pits are so safe. So if you don't fall into it. So one thing is that we could identify this individual by dental, and also we confirmed the identity by a plate. And we could see all these fractures. There's a thermal alteration, but we have to distinguish if all the fractures that we see are because of the fire exposure, or do we have any other fractures? And we could see, especially this fracture here, we didn't like it very much. So it was something that it didn't fit very well with the fractures by the fire. So if you remember that hand draw map, we could see that there were some bricks here, and that could be attributed most likely to a breakage or a fracture because of the fire. We don't know, however, if it's it was perimortem, so that was the first hit, and then it was altered in the fire, or it was something everything in between. Because for us, it's very difficult to differentiate in the timeline of the perimortem. For us, the perimortem timing, it's very wide in forensic anthropology, because we assess it not by inflammatory reaction or so on like pathologies, but we assess it by how the bone looks like. And so the blunt force trauma in the right lateral cranium was distinguished from the thermal trauma, and that was very important. And without the hand draw map, that would be impossible, absolutely impossible to connect. So again, how important is the recovery? And I just wanted to thank you. I normally love to be in the morgue to process the remains and do the skeletal analysis, but I can assure you, since I'm here, I have learned from Dr. Dirkmatt, if you don't do a proper recovery, that won't be very, you cannot have, or you cannot provide all the information that you can provide if you don't record the scene and then integrate this information into the case. Thank you so much. If you have any questions, just feel free to ask, and that's my email in case you need it ever. I hope you never need us, because that would be a very bad sign, but.
I have two questions. When there's a gunshot wound, does it always go straight, or is there different parts of the body that can cause it to not, if they're,
No, sometimes, so it depends on where we are. So the structure, the morphology, and the structure of the bone, how it's the spongy bone, how it's the cortical bone, how this is distributed in the bone, and the overall shape makes a lot of difference. So the best examples for gunshots, it's the cranium, because it's not something that it's raw, it's like we have lots of flat bones that we can see the beveling very well and all that. So in the cranium, so the trajectory can be the bullet can be inside the cranium, or the bullet can have like an entrance and an exit. So, and same thing with, like in the long bones, normally you have like a big destruction, so you don't have like a beautiful, beautiful entrance or exit as if you have it in the cranium, and that's why, like, always that I always say to my students when I tell you about like entrance, exit, think about the cranium, and then we will apply it to other parts of the skeleton. So each bone is very different, but the best examples are in the cranium.
And then just another question, since you specialize in dental, if you have remains that have been left outside, how long do the teeth stay intact? Because sometimes it looks like some of them have fallen out. Is that over a long period of time?
So there was a study that wanted to relate like the postmortem interval according to how many teeth were fell, like postmortem. I think that's not a good. It's not a good method, because I mean it's true that like the longest that we have someone out there, the most possibilities that we have to lose teeth, especially the single rooted teeth, but also that this person had periodontitis, that we have like loosened teeth, and then you will lose them like much earlier. So also if you have root resorption, so there are a lot of factors that can affect that. So I don't think that would be a very good. I wouldn't use it.
Yeah.
All right, everyone, thank you very much. Our session is now over. We do have a poster session starting at 6 p.m. if anyone wants to join us for that later.